Hello, am I audible now? Can you hear me now? I hope you can hear me now and see me well. All right. All right, guys. So now you can hear me and see me. So today we are going to do a rapid, quick revision of microbiology. I've taught you all the things in the great lectures, right? But this is going to be a quick revision. That is what is going to be there just before the examination. What are the important key points that you need to see? We are going to discuss about all those things. The only important thing that I want from you guys is to be interactive. Just try to answer the things that you are going to see, right? So this is going to be a very quick and crisp revision of microbiology, where I will be telling you all the important points which can come in your examination on 28th of June, right? So these are going to be the things, right? So let's start with the first topic that we are having is going to be about the microscope. So what is going to be a microscope and who is this particular gentleman? Try to answer. This particular gentleman is your Antony von Leeuwenhoek. What is the name of this person? Antony von Leeuwenhoek. He discovered the microscope. The first microscope is discovered by this particular person, right? So the next question is, how many types of microscopes do we have, right? This one is going to be your light microscope. This is what you see very commonly in your laboratories. This is going to be your light microscope. This is your light microscope. So they are going to ask you certain questions about the parts. In the last few exams they have asked some questions about the parts. So this is going to be your eyepiece. Through eyepiece you are going to look from this particular area. This is going to be the nose, and these lenses are going to be your objective lenses. These lenses are going to be your objective lenses. The next question they are going to ask you is going to be, how many types of objective lenses do we have, right? So we have got four important type of objective lenses here with the power of 4x, 10x, 40x and 100x, right? These are going to be the power for objective lenses, and the power for eyepiece is going to be what? That is going to be 10x, right? So this 4x we are going to use it as a scanner. For what purpose we are going to use 4x? For the scanning purpose, right? So this is going to give you 4 times magnification. 10 times magnification we are going to use as a low power. This is going to be low power. 40x is going to be what? High power microscope. High power microscope you are going to get, and at 100x you are going to get the oil immersion field. And remember, oil is going to increase the refraction and you are going to get the better picture, right? So this is going to be what? This is going to be your simple light microscope. Another question they have asked in the past is going to be, what is going to be the source of light for this microscope? So the source of light for this microscope is going to be what? Transmitted light. What kind of light is going to be the source of light? Transmitted light is going to be the source that we are going to use, right? The session will be approximately 2 hours, where I will be covering all the important points of microbiology, right? Okay, so this is going to be your normal simple microscope. This is your binocular microscope. This is your these are your uh, two eyepieces through which you are going to look at the picture. This is going to be your binocular microscope, right? What is this kind of microscopy? This is your exam question. This kind of microscopy is known as what? Can you appreciate? The background is dark, so this type of microscopy is known as dark ground microscopy. Dark ground microscopy. They can give you picture. They can give you picture like that, and they can ask you which is the kind of microscopy we are having. कौन सी microscopy है? So this is going to be your dark ground microscopy, and the organism is going to be, this is your spiral like organism. This spiral like organism is going to be what? This can be a spirochete, it can be Treponema pallidum, for like which is going to cause what? Syphilis. So this is going to be the one they can ask you, right? So this is going to be dark ground microscopy. What is the source of light we are going to use? R for R, you can remember, this is going to use what? Reflected light. The source of light here is going to be what? Reflected light is going to be the source of light, and the most important thing here is going to be what? what is a special feature through which we are going to get this kind of images. That means background is going to be get black and the organism is going to be shining. That is because of a special structure in your microscope, and this is going to be your central aperture. This is going to be your central opaque area. Central opaque area or central aperture is going to be there, which is going to cause this only the organism is going to get shine up, right, and remaining background is going to get black, right. Another microscopy, we are having this big structure. This is going to be your, yes, this is going to be your electron microscopy. This is your electron microscopy. The last exam they have asked, NEET PG exam, they have asked what is going to be the fixation for electron microscopy. So if they ask you this question, what is going to be the fixation for electron microscopy, the fixation is going to be what? How you are going to fix the slide? This is going to be with the help of 2 to 2.5% glutaraldehyde. With the help of 2 to 2.5% of glutaraldehyde, and this is also known as what? Sidex. This is also known as Sidex or Sidex, which is used as a disinfectant, right? This is going to be used for what purpose? To sterilize what? Your scopes. To sterilize your scopes that we are having, internal scopes. These are going to be sterilized with the help of this glutaraldehyde, and this is the same glutaraldehyde which is going to be used as a fixator in electron microscopy. What is going to be the scientist who discovered electron microscopy? The name of the scientist is going to be what? E for E, that is going to be your Ernst Ruska, right? Electron microscopy was discovered by Ernst Ruska. An important thing, whenever you will find black and white image, like maybe you are looking at your mitochondria inside your cell, or maybe any viruses, anytime you are going to get black and white image, this is going to be due to what? Electron microscopy. This is going to be due to your electron microscopy, right? So this is going to be your electron microscopy. Okay, what is this type of microscopy that we are having? Total we are having 5 types of important microscopes that you need to remember. This particular one is going to be known as what? This is known as fluorescent microscopy. This is your fluorescent microscopy, and the dye that we are going to use here is known as what? The chemical that we are going to use here, that is going to be the fluorophore. We are going to use here is going to be the fluorophore. The name of the chemical is going to be fluorophore, right? This is going to be the microscopy that we are having. We are using for what purpose? For the screening purpose. What is the principle of this particular microscopy? What are the special things about this microscopy? This is important one. They can ask you question from here. The principle is going to be what? This chemical is going to convert the lights of shorter wavelength into longer wavelength. So the chemical is going to convert the light of shorter wavelength into longer wavelength, and that is going to be visible to you. So if the organism is present, that is going to be shining up, right? That is going to be shining up, right? Another important thing, what kind of mirror is going to be there here? Special mirror is going to be there, that is known as dichroic mirror. Dichroic mirror is going to be there, and important thing, the source of light is going to be what? UV light. UV light. In INI-SET they have asked you this UV light is coming from which lamp? That is going to be coming from your maybe xenon lamp, right? So this is going to be what? The UV light which is coming, which is reflecting through the dichroic mirror, and it will go to the structures. If the organism is present, what will happen? Shorter wavelength lights will convert into longer wavelength, and these are going to be visible through your eyes, and this is how you are going to visualize this particular structures, right? Some common examples they are asking you, right? What kind of dye we are using for what particular purpose, right? So for example, if they are asking about TB, what is the name of the dye for TB? Try to answer. What is the name for dye for TB, right? For malaria, beta, for malaria, to identify your fungus, what are the dyes we are using? For TB, the dye that we are using is going to be what? That is going to be auramine. Yes, auramine, rhodamine. Auramine, rhodamine is the dye that we are using for identification of tuberculosis. So basically we are putting this dye around the vessel, right? And now what is going to happen? You are going to put your organism, and this dye will be having what? Fluorophore chemical. So what will happen? If this dye will be having these organisms present, right? If the sample is going to have these organisms present, what will happen? Automatically, because of this UV light, because of this dichroic mirror, what will happen? The light of shorter wavelength will convert into longer wavelength, and these organisms are going to appear shiny to you, right? And this is how you can appreciate that this person might be having what? TB, right? This is for the screening purpose we are going to use, right? What is the dye we are going to use for malaria, beta? For malaria, we are going to use what? That is going to be your acridine orange. Acridine orange for malaria. And F for fungus, this is going to be your calco-flower white. For fungal, we are going to use what? Calco-flower white. So these are going to be the important dyes that we are having for TB, malaria, and fungus, right? This is going to be the thing, right? Now the last type of microscopy we are having is phase contrast microscopy, right? What is this phase contrast microscopy having important features? It is also going to be a type of light microscope only, right? But the important feature is going to be what? It is going to have what? These annular rings are going to be there and phase plates are going to be there. So two important entities you have to remember here. It will be having what? Annular ring, that you can appreciate. Annular ring is going to be there. This is going to be ring like structures are going to be there, which is going to be completely surrounding this particular area. This is going to be your annular ring, right? And here you will be having what? Phase plates, which is going to produce some changes, and because of that, what is going to happen? The organism is going to appear moving. Organism is going to appear moving, right? This is what is going to cause you. This is going to give you maybe 3D pictures you can get, maybe motility you can identify for the organism. So this is due to what? This is due to phase contrast microscopy, right? So how many types of microscope we are having? Light microscope, we are having dark ground microscopy, electron microscopy, fluorescent one, and phase contrast. If they ask you any question, you are able to answer. These are the important questions they have asked from this particular topic. 9. Okay, you understand? Speed is okay. Should I increase some or decrease some? Right? The next thing we are having is about the genetics, right? What is this genetics of a bacteria, right? So what is the most common method of genetic transfer? What is the most common method of genetic transfer or resistance transfer? So the most common method, if they ask you, most common method is going to be what? This is going to be conjugation. This is going to be what? Conjugations, right? This is going to be conjugation. And what is conjugation? When a cell will combine with another cell with the help of pili. So whenever this combination is happening with the help of pili, this is known as what? Conjugation. And this conjugation is going to be the most common method of genetic transfer, right? It can transfer the resistance between the bacteria. Anything, if they ask you the most common method, the answer will be automatically going to be your conjugation. But if they ask you what is this transformation? What is this transformation? Transformation means this bacteria is going to take up this free DNA material from the environment, right? This free DNA material is going to be taken up by the bacteria. This is known as what? Transformation. And this free DNA material is going to be taken up by the bacteria. This is known as transformation. Which is the bacteria which is going to show you transformation? Try to answer. This is going to cause, this is known as what? Transformation. Taking up new DNA material and involving into the same particular bacteria. This is known as what? Transformation. The most common bacteria showing transformation is going to be your pneumo. Pneumo means air. Coccus means it is going to be a round bacteria. So this is going to be pneumococcus, right? Can you tell me what is this pneumococcus is important, right? It is a capsulated bacteria. Just remember 2-3 points about this pneumococcus. It is the most common cause for meningitis, adult patient. It is the most common cause for pneumonia also, right? So in typical adult patient, community acquired pneumonia, any person is going to get pneumonia. What is going to be your answer? Pneumococcus, right? Another person coming to you, young adult person coming to you with the symptoms of what? Meningitis, fever, headache, nuchal rigidity, Karnic positive, Brzezinski positive. This is going to go in the favor of what? Pneumococcus. Yes, this pneumococcus is also known as streptococcus pneumonia. And what kind of pneumonia this particular one is going to cause? This is going to cause your lobar pneumonia. Lobar pneumonia. Okay, beta. This is going to cause your lobar pneumonia. So this is going to be the transformation, right? What is transduction? Transduction means this particular entity is what? This is a bacteriophage, right? So whenever a bacteriophage is going to be involved, what is this bacteriophage? It is going to be a virus, right? And virus has got a special capability. Just remember one thing: virus have everything. Virus has got everything, but virus cannot replicate on their own, right? They do not have what? Mitochondria. They do not have what? Mitochondria, your DNA replication organelles, ribosomes, all these things are going to be what? Not working properly, right? So that is the reason why this bacterias are going to, sorry, this virus is going to release their genetic material into the other cells, right? So ये virus हमेशा दूसरे में आके replication करेगा, right? So virus is coming into your body just for replication. Virus is infecting bacteria just for replication, right? So this virus is going to come, it is going to transfer the genetic material, maybe DNA, RNA, right? It is going to come into the cell, and now the cell is going to do certain process. What are going to happen? What are the processes which are going to happen? So this is going to be the genetic material. Let's say this is the genetic material is going to be RNA. This RNA will enter into ribosomes beta, and ribosomes are going to do what? Sorry, this RNA will combine with the nucleus, and it is going to make what? mRNA, and now this mRNA will enter into your ribosomes, and ribosomes has got a capability to produce what? Proteins, and these are going to be the proteins which are going to come out, and these proteins are going to further make what? They will enter into your Golgi apparatus. All those things will happen, and ultimately you will get a new replica of the same virus, right? So this is known as what? Transduction. This is how viruses are going to get replicated, right? So this is going to be happening with the help of bacteriophage. Bacteriophage is nothing but a virus which is going to infect the bacterias, right? And this is what is going to happen in the most commonly seen with the streptococcus. Sorry, most commonly it is seen with the staph aureus. So the most common entity which is going to show you this process is known as what? Transduction. This process is known as what? Transduction. So whenever a bacteriophage is involved, this is known as transduction, and the most common method of transduction is seen in staph aureus. It is a constant fight between staph aureus and us that is happening for such a long time. Earlier, staph aureus was definitely killed with the drugs like penicillin, right? Then staph aureus has developed something, right? It has developed something which is going to, it has developed some penicillin binding proteins, and then this penicillin is not working on the staph aureus. Because of this penicillin binding proteins, this staph aureus is not going to killed with the penicillin. So now we have developed what? We have developed the methicillin, right? The drug that we have developed is methicillin, right? And now this methicillin is going to get resistant. This bacteria is developed resistant because of what? Because of transduction, right? So now this has become what? MRSA. What is the drug of choice for MRSA? Methicillin resistant staph aureus. What you are going to give? This is going to be your vancomycin. And now what happened? Nowadays what happens? This vancomycin resistant staph aureus is going to be there, and the drug of choice is going to be what? This is going to be your daptomycin, or we can also use what? Linezolid. We can also use what? Linezolid, right? For pulmonary infection, we are going to use linezolid, since it is daptomycin is not working in the lungs, right? So we can use linezolid. We can also give daptomycin for MRSA infection, right? So this is going to be what? This is going to be the genetic methods that you need to remember. First one is conjugation, second one is transformation, third one is transduction. So if they ask you most common method of genetic transfer, answer will be conjugation, right? If they ask you the method of genetic transfer which involve free DNA material, this is going to be what? Transformation. What is going to be the important organism? This is the most common cause for pneumonia and meningitis. This is going to be your streptococcus pneumonia, right? If they ask you if a bacteriophage is involved, this is known as what? Transduction, and the organism is going to be what? Staph aureus. That is going to show you the most common organism which is going to show transduction. The answer will be staph aureus, right? This bacteriophage are going to be dangerous. They can also cause what? Lysogenic conversion that we have studied, right? They can make a non-pathogenic bacteria into a pathogenic one, that is known as that is known as lysogenic conversion. Okay, the next important topic is about the physiology of a bacteria, where we will learn about the how a bacteria is going to get grow in the life. So this is going to be the life cycle of a bacteria. First phase is going to be what? Leg phase. First phase क्या होता है बेटा? Leg phase. This is going to increase in the size. Just look at the humans also. We are increasing in size first, like 1 year, 2 year, 5 year, right? Then they are going to get married, right? And the bacteria is also enter into the log phase. They will also get married. And now you will be having what? Increase in the numbers, right? Now you start to have what? Divisions. Reproduction will be there, right? And because of the divisions, you are going to increase in the number. And then what happens? This is going to be the stationary phase, where the number of live bacteria is going to be equal to the dead bacteria, right? Dead bacteria, right? So this is going to be the stationary phase, and ultimately you will start to have what? Decline phase is going to be there. The bacteria start dying. So there are going to be 4 important phases in the life cycle of a bacteria. What are these? First one is A. This is going to be the first phase. Log is going to be the second phase. Stationary phase is going to be the third phase, and lastly we are going to die. This is going to be the fourth phase, right? So let's try to understand what questions they are going to ask you from this particular part, right? So the first one we are having is leg phase. What happens in leg phase? Sir, just like humans, it is going to increase in size. Size is going to get increased. Metabolic activity is going to get increased, and they will prepare for the marriage or division, right? This is going to be what? Leg phase. What happens in the log phase? Log is going to be what? Exponential phase, and now post marriage you are going to have the division, right? So rapid cell division is going to be there, right? And growth, this is the most metabolically active phase. And most important question, this is the best phase for what? Antibiotic action, right? When the bacteria is dividing more, your antibiotics like static antibiotics or maybe cidal antibiotics will be working very nicely here, right? The first question is going to be antibiotic action is going to be get when you are going to get the best antibiotic action, this is going to be the log phase, right? Third one is going to be your stationary phase. What happens in stationary phase? Here you will be having what? Nutrient depletion and waste accumulation, right? Growth rate is equal to death rate, right? And here you will be having what? What is stab? Post marriage you will be getting stabbed, right? What happens? Stabbed. As for first thing is going to be what? As for sporulation. First thing is going to be what? As for sporulation, right? So the examiner has asked the question, sporulation is going to happen in which phase, right? So this is going to be your phase 3 of the bacterial growth curve, right? So sporulation is going to be there. Another question they have asked in ionized set examination, the spores are going to be resistant due to which acid? Spores are resistant, they are heat resistant due to which acid? My dear doctors, try to answer which acid is going to cause the spores heat resistance, right? What is going to be your answer, right? T4, second thing, it is going to make what? Toxins, right? We know about 2 types of toxins. We are having exotoxin and endotoxin, right? So bacteria is going to produce some toxins. These are going to be your exo and endotoxins. A for antibiotic production, antibiotic production to kill other bacteria, and B for bacteriocin production, bacteriocin production, right? They are going to make what? Bacteriocin, and this bacteriocin is going to inhibit the growth of other bacteria. It will inhibit growth of other bacteria. What is the role of this bacteriocin? It is going to inhibit the growth of other bacteria, right? Very good, Mahesh, right? This is going to be what? This is going to be D-picolinic acid. Dipicolinic acid is going to be there, which is going to make these spores heat resistant, right? So these are going to be your sporulation or spore formation is going to be there, right? The next phase is going to be your death phase. Death is going to be the decline phase, where everything, this bacteria will start to die, right? So this is what is going to be the life cycle of a bacteria. They have asked some questions from here. So this is going to be the first phase. First is going to be which phase? Lag or log? Just remember, A is going to be first, lag phase is going to be there. Maximum increase in the size is going to happen. The person is going to get bigger, right? 10 saal, 12 saal, 13 saal, till 21 or 18, this person is going to get bigger. Second phase is going to be your log. Now this person will get married, right? And now you will be having maximum number of division, and whenever there is going to be maximum number of division, you can have the antibiotic action. Best antibiotic action is going to be there. Third में post marriage you will be having dhoka, so this is going to be what, stab is going to be there. S for sporulation is going to be there. T for toxin production, A for antibiotic production, and B for bacteriocin production. What is this bacteriocin? Another expected question. This bacteriocin is going to inhibit the growth of other bacteria, यानी ये दूसरे को नहीं grow करने देगा, he himself will live here, right? So this is going to be the stab, and lastly we are having a death phase. This is the important topic we are done with, right? So this is going to be the bacterial growth curve, right? Now the next topic we are having is about the motility of a bacteria. What is the motility of a bacteria? Try to answer this. Tumbling motility. कौन करता है tumble? Those people who are alcoholic people, they will drink and then they will tumble, right? So this is going to be seen with the listeria. Listerine में भी क्या होता है बेटा? Listerine will also have what? Alcohol, somewhat alcohol. It is a mouthwash, right? So this is how you can remember. Listeria will be having tumbling motility. Tumble करे, right? आप लोगों ने एक गेम खेला होगा डार्टिंग, राइट? सो द डार्टिंग इज गोइंग टू बी देयर, राइट? वीसीडी वी हैव रिमेंबर्ड, वीसीडी होता था सीडी से पहले, डीवीडी, वीसीडीज, राइट? सो दिस इज गोइंग टू बी डार्टिंग इज सीन विद विब्रियो कॉलेरा and Campylobacter jejuni. Campylobacter jejuni, right? So we darting motility is seen with Vibrio cholera and Campylobacter jejuni. If I want to mention some important points about these bacteria, some important lines you can remember. So Listeria, remember the story of Lisa auntie. She went to the market and she will be having what? She will be having premature rupture of membrane can happen. It is happening, this thing can happen, and this bacteria lives in a cold environment generally. That is why we are telling our females not to eat directly from the fridge. Pregnant females, right? They can have what? Premature rupture of membranes. Maybe they can have what? Abortions can also happen. And in the adult patient, in the children's, it can cause what? Meningitis. Adult older patients, it can cause what? Meningitis. So these are some important points about this listeria. Lisa auntie you can remember. So Anton test was positive, remember? Keratoconjunctivitis in the eyes of rabbit, right? So Lisa auntie wali kahani, if you can recall, right? This is going to be what? Listeria, right? What about this Vibrio cholera? It is going to cause you a diarrhea. What kind of diarrhea? This diarrhea is known as, yes, this diarrhea is known as rice watery stools is going to be there because of this Vibrio cholera. Important question from the in the examination, they have asked you, where does this cholera toxin is going to bind? I am focusing on the PYQs. So this cholera toxin is going to bind on the GM1 ganglioside. Very important, right? We cannot read each and every bacteria, but I can give you the most important information that can be useful, right? Earlier they have asked this question, where does this Vibrio cholera is going to bind? This is going to be GM1 ganglioside. In the last exam NEET PG, they have also asked you one more important question, what is going to be the fragment, right? So just remember where exactly this is going to be your fragment B. Very important question anticipated, right? They can ask you from other subjects also, right? This is going to be the fragment B of GM1 ganglioside, where this cholera toxin is going to bind, and this cholera is going to cause what? Rice watery stools in the patient. Very important thing, right? Campylobacter jejuni question also they can ask you. Remember about the Pune outbreak story, right? So this Campylobacter jejuni has got some associations with the syndrome. This syndrome is known as Guillain-Barré syndrome, and the treatment of choice for Guillain-Barré syndrome is going to be what? Intravenous immunoglobulins, right? So this person can have ascending paralysis because of this Campylobacter jejuni, right? This Campylobacter jejuni can cause dysentery in the patient. Patient में diarrhea होगा, dysentery होगा, limb weakness आएगी, right? After weakness, this person will be having ascending paralysis, right? This is going to be a diagnosis for GBS, right? And post GBS, you can use this IVIG and the patient will be fine, right? So these two organisms are going to show you what? Darting motility. Swarming motility is seen with what? Swarming motility is seen with the, swarming का मतलब क्या होता है? Spreading. Swarming मतलब छा जाना या spread हो जाना. This is seen with the Proteus. This is seen with the Proteus, right? This is the swarming motility. Can you appreciate? ये पूरी slide के ऊपर छा गया, right? So this is known as swarming motility. This is seen with the Proteus organism, right? What about Proteus? Important thing, this Proteus is going to cause some kind of a stone. What is the stone it can cause? It can cause true white stone. It can cause true white stone or staghorn calculi, which are going to be radio opaque. That means they can be visible on the X-ray, right? So this is going to be your true white stone that can happen, right? And you can also find this protease antigens can cross react with the rickettsial antibodies, and this reaction is going to be there. This reaction is Wheel-Felix reaction, right? Wheel-Felix test or Wheel-Felix reaction can be there, where the antigen of protease can cross react with the antibody of rickettsial organisms. This is an important question they have already asked, right? So this is about the protease, two important points about the protease, right? Slow form of swarming is seen with the Clostridium bacteria. Slow form of swarming, stately motility also you can call it, right? This is seen with the Clostridium. We know about Clostridium tetani can be there. We can have Clostridium tetani can be there. We can have Clostridium perfringens, right? So perfringens, I've also taught you this thing in FMT, right? This perfringens is going to be responsible for producing an enzyme. This is your lecithinase, right? Lecithinase, and lecithin is a lipid, so it is going to break the lipid. It is responsible for the putrefaction post death of a person. This Clostridium perfringens will be there. It can also cause food poisoning. Tetany, you already know this tetany can cause what? Tetanus bitter. This can cause what? Tetanus, right? Clostridium tetani, right? So this is going to be Clostridium, right? Falling leaf, patta, you can remember like that, patta giradia. So falling leaf motility is seen with giradia or giardia. Giradia or giardia is sounding like similar, right? So this is going to be Giardia lamblia, which is the most common parasitic infection that can happen in the intestine, right? So this is going to be your Giardia lamblia. It is also referred as the great grand old man of GIT, right? So this is going to be the Giardia. How many pairs of flagella you are going to appreciate here? This is going to have 4 pairs of flagella, or total is going to be what? Total 8 flagellas are going to be there. So this is important. Only two important flagellate organisms you can remember. One is going to be this particular one. Another one is going to be Trichomonas vaginalis, right? So this corkscrew motility is seen with Treponema pallidum. Treponema pallidum, and this Treponema pallidum is going to cause what? Syphilis, right? This Treponema pallidum is going to cause what? Syphilis, right? Syphilis. Yes, this is a trophozoite of Giardia. Trophozoite of Giardia. Okay. लाशिंग, लाश को कैसे रखोगे बेटा? लाश याद रखो. लाश को कैसे रख रहे थे हम लोग? बोरे में लपेट के, राइट? सो दिस इज गोइंग टू बी सीन विद द बोरेलिया and leptospira. This is seen with two organisms, Borrelia and leptospira will be having lashing motility. लाश को कैसे रखोगे? बोरे में लपेट के. So you can remember बोरे में लपेटना, right? This Borrelia burgdorferi we are having, right? And we are having leptospira. Leptospira is important, right? What are the important points about leptospira? Try to mention, right? Leptospira question they are asking very commonly. What are the important points? Try to mention. Then I'll give you some insight about this leptospira. It is a hot topic for examination. This leptospira they are asking from 2-3 exams, right? Next is going to be T for twitching, T for Trichomonas vaginalis, right? Trichomonas vaginalis, right? And remember this vaginalis is having V, and V is looking like 5. So how many flagella we are having here? Total 5 flagellas are going to be there. So this is going to be an organism which is going to be Trichomonas vaginalis, right? And this Trichomonas vaginalis will be having these 5 flagellas you can appreciate, and this is also going to cause what? Your strawberry vagina, strawberry cervix, right? Colpitis macularis, you can remember Colpitis macularis, strawberry cervix, right? This is going to be your Trichomonas vaginalis. The drug of choice is going to be what here? Okay, roti कैसे बनाते हो? roti, you can remember roti you are going to make with the help of balan, right? So this is going to be your Balantidium coli. Balantidium coli is going to be there, which is going to have what? Rotatory motility. It is the only ciliate organism that you have to study, right? So this is going to be your rotatory motility seen with the Balantidium coli, right? Another exam question they have asked you is about the differential motility. Differential motility is going to be seen with one important organism. Is going to show you two differential type of motilities. What is that organism? This is going to be again Listeria beta. Listeria is going to show you two types of motility. It is going to be showing you sometimes it is going to be motile, sometimes it is going to be non-motile, right? So you can remember Listeria was the alcoholic drink that we are talking about, right? So it can show you two types of motility, motile and non-motile. So when you are young, you sit at home or drink, or you go out and then drink, right? So when you are young, obviously you go out and then you drink, right? This is going to be at 25 degrees Celsius, right? At 25 degrees to 27 degrees Celsius, or maybe 25 to 27 age, you can remember, this person is going to be drinking outside, right? So this is going to be a motile person. But when you will get older, right, at 37 degrees Celsius, right, or 37 age, you start sitting at home and then you start taking your packs, right? So this is going to be when you become older, you are going to have, you become what, non-motile. When you are young, you are going to be motile. And what is the use of this particular thing? When this Listeria will enter into your body, 37 degrees is going to be what, your body temperature, right? So this is going to be the thing where it will become what, non-motile. But the disease it can cause, right, definitely it can cause what, diseases, right? So Listeria question has been asked previously. It is important cause for two important things, at least you have to remember. Two important things I can mention here. First one is going to be what, premature rupture of membrane in the females, right? In the females, this Listeria can cause problems, right? And second important thing is going to be what, in the in the older patients and in the young individuals, it can cause what, meningitis. It can cause what, meningitis. So it can cause meningitis in the elderly patients, in the young patients, right? And it can also cause what, premature rupture of membrane. So these are the two important points that you can remember, right, about the Listeria. Yes, Leptospira is going to be there. Yes, Ruby is telling all the story. Lisa auntie Palkam Bazaar mein gayi with premature rupture of membrane because she ate cold food from the fridge, right? So that was the story we have seen, right? Again, this particular one that we are talking about, Leptospira is seen with three triple R, you can remember. These are going to be what, this leptospirosis. They are asking some questions about this leptospirosis. Always remember some keywords are going to be there, right? They are going to have differential diagnosis with the scrub typhus. Your like you can say your scrub typhus can be there. You can have dengue, right? You can have brucella, right? All these things are going to look similar because the patient will be having fever is going to be there. All the symptoms, body pain is going to be there, right? But the important keyword is going to be what, some keywords you have to remember, and that is going to be your conjunctival suffusion is going to be there, right? So this conjunctival suffusion word you will find, right? Urine, rice field, paddy fields are going to be there. You think about this particular one, right? So this is going to be what, Leptospira. And we are having these motilities are going to be there. I've given you some information about these organisms also, right? So tumbling is going to be seen with the Listeria also going to show you differential motilities. Darting with the VCD, swarming with the Proteus. This is going to be the swarming of Proteus. Slow form of swarming is seen with the Clostridium. Falling leaf, Patta giradia. This is going to be Giardia lamblia. Corkscrew motility is seen with the Treponema pallidum, going to cause what, syphilis, important one, right? Lashing motility, बोरे में लपेट के लाश को रखोगे, right? T for twitching, T for trichomonas, right? R for rotatory, roti कैसे बनाओगे with the help of balun, right? And differential you already know. So these much of motilities at least you have to remember. Now we will discuss about certain food poisonings. What are the causative agents for these food poisonings, right? Because nowadays they are not asking you direct question what is going to be the organism that is going to cause this, right? Rather they will give you some history and they are going to ask you some question. That means they will give you history. A person went to a party and he ate something, or maybe he had fruit, cake or something. They will mention in the keywords, right? And then they will give you, the patient will start to have what, vomiting, sometimes diarrhea, sometimes fever, right? So we need to identify what kind of vomiting, what kind of diarrhea, and what kind of bacteria is going to be involved in, according to their incubation period, right? So what are the different kind of food poisonings that we can have, and what are the product which are associated, we are going to mention here, right? So the first one we are going to have is Bacillus cereus, right? So Bacillus cereus is going to have two types of strains, right? Bacillus cereus is going to have two types of problem. There are going to be two things. One is going to increase the cyclic. I told you this thing in the lectures. You can just recall. I'm not going in detail. You can have cyclic GMP activation. You can have cyclic AMP activation. Whenever is going to be there cyclic AMP activation, the person will start to have what? Diarrhea, right? And cyclic GMP is going to increase your nitric oxide. Remember, your achalasia cardia also has got the same pathology, right? So this is going to be cyclic GMP, which is going to increase nitric oxide, which will relax all the muscles. Your esophageal sphincter will get relaxed, and what will happen? The content of the stomach will come out, right? So this is going to be what? Causing your vomiting, right? So cyclic GMP will increase nitric oxide and leading to what? Relaxation of the sphincters, leading to what? Vomiting in the patient, right? And cyclic AMP, you can remember, it is going to deal with the water absorption, or also you can remember A for NS, right? So this is going to be something which is going to cause what? Diarrhea, right? So Bacillus cereus has got two important things, right? Very good, your incubation period is correct, right? You told me 1 to 6 hours is going to be there, right? So but two important products, two important type of problem we can have emetic strain, we can have diarrheal strains of Bacillus cereus, right? So emetic strain is going to be there, diarrheal strain is going to be there. The emetic strain is going to be, remember, activating your cyclic GMP and Ghoda GMP. You can remember G for Ghoda, and Ghoda kahan rehta hai beta? It is going to be living in the astabal. So you can remember it is going to be heat stable. It is going to be what? Heat stable toxin, whereas this diarrheal one is going to be what? Heat labile, right? So if you are going to have this reheated Chinese fried rice, one toxin is going to be heat stable, मतलब गर्म करने से भी नहीं मरेगा. दूसरा टॉक्सिन हीट लेबाइल होता है. अगर हीट किया तो ये डायरिया नहीं होगा, बट ये वोमिटिंग हो सकता है पेशेंट में, राइट? सो वी आर गोइंग टू हैव हीट स्टेबल एंड हीट लेबाइल टॉक्सिन. ऑलवेज रिमेंबर, जब भी हीट स्टेबल टॉक्सिन की बात होगी, यू विल थिंक अबाउट व्हाट? घोड़ा. And this Ghoda, G for Ghoda, you can remember stable is going to be with the Ghoda, right? It will activate cyclic GMP, will lead to activation of nitric oxide, and that will cause vomiting within how much time? Within 1 to 6 hours is going to be the incubation period, right? When it will happen? After eating reheated Chinese fried rice. After eating what? Reheated Chinese fried rice, right? So what is going to be the history you will get? Some person is going to go out and had a reheated Chinese fried rice. What is going to be the mechanism also you understand, and what will happen within 1 to 6 hours is going to be the vomiting in this particular person, right? This is going to be what? Emetic strain, and this is going to cause what? Bacillus cereus. What about this diarrheal one? The sorry, the incubation period for diarrheal one is going to be what? Different. This is going to be 8 to 16 hours, and it is going to activate your cyclic AMP, leading to what? Diarrhea in the patient. Person will have what? Diarrhea, right? So you can remember cyclic GMP and AMP activation can be there. Bacillus cereus, if they are asking about the vomiting symptoms, you need to think about the emetic strain. Incubation period is 1 to 6 hours. If they are talking about the diarrhea in the patient, this is going to be 8 to 16 hours is going to be the incubation period, right? And this is going to be what? This is going to be diarrheal strain, right? What about the Staph aureus? The Staph aureus is associated with what? The Staph aureus is associated with meat and milk product. They can write you pastries, they can write you chicken curries, right? They can write you anything related to meat and milk product. This is going to be within 1 to 6 hours is going to be the incubation period for this Staph aureus. Person is eating something in the night, and in the morning he will start to have what? Bouts of vomiting. So this is going to go in the favor of what? Staph aureus, right? The next one is Clostridium botulinum, right? This is going to be important one. This botulinum toxin is going to be this Clostridium, we already know this Clostridium is a aerobic bacteria, anaerobic bacteria. It is an anaerobic bacteria. Remember, the mnemonic fuso, cannot breathe air, right? So this is going to be the cannot breathe air. They cannot survive in the, these are the anaerobic bacteria. They cannot survive in the oxygen. So C for what? Fusobacterium. This is going to be Clostridium. B for bacteroids, and A for actinomycetes, right? So this C for Clostridium is a anaerobic bacteria. It does not like oxygen, right? It will die in the oxygen, right? So this is going to be what? This botulinum is going to be present in the canned food. We already know this canned food do not have oxygen, right? वो जो डब्बे आते हैं उनके अंदर ऑक्सीजन नहीं होता है, right? So if we are eating those canned food, you know what infection we can get? We can get the infection from the Clostridium bacteria, right? This Clostridium bacteria can be there, right? And it has got some important toxin. The name of the toxin is going to be what? C2 enterotoxin. C2 enterotoxin. And this is an expected question because this is a toxin which can cause what? It is a kind of food poisoning, but this toxin is going to cause what? Constipation. And other symptoms are also going to be very different, right? Symptoms are going to be patient will start to have dysarthria, patient will start to have diplopia, double vision will be there, dysphagia will be there, right? Constipation will be there. And these are all what, you know, these are things which are going to be coming under the food poisoning. This is happening because the person has eaten something from the canned food which was infected with the Clostridium bacteria, right? So this is going to be the canned food, and this is going to be what? This is an important one. You can remember the food poisoning. Generally we think about vomiting, diarrhea, but this is a food poisoning which is causing what? Constipation. Important. This is going to be your C2 enterotoxin that can be there, right? What else you can get? This Clostridium botulinum you can also get from the honey, and that is too with the raw ingested honey, raw honey ingestion. And generally this thing is happening in the babies, you know, mothers will give this honey to the babies, right? And what will happen after taking this particular honey? This baby will get the spores of this Clostridium and they will get activated. What this Clostridium will do? This raw honey will cause what? They will inhibit the, how they will be working. Let me just give you a brief insight. They will enter into your cell, they will enter into your inside the neurons, right? And what will happen? They will inhibit the snare protein. This is your another question. What is the snare protein is useful for? They will inhibit the cleavage of snare protein. The snare proteins are responsible for what purpose? They are responsible for exocytosis. Exocytosis. So suppose this is your neuron, and if we are having Clostridium botulinum or botox injection or botox botulinum toxin, anything is coming into your body, what will happen? This acetylcholine vesicle is not able to come out, right? They will not able to do the exocytosis. Just think about it. If this acetylcholine is not going to come out, do you think there will be further movement or contraction or anything is going to happen? No, right? So this is how this botox, you can say botox people are doing, or this botulinum toxin is working. They will inhibit the snare protein, right? And snare protein has got a function of exocytosis. Exocytosis nahi hoga, right? So, and this thing can lead to, in the baby will have flaccid paralysis, and this is known as floppy infant syndrome. Your pediatric integration, this is known as floppy infant syndrome, or flaccid baby will be there. Floppy infant syndrome or flaccid baby will be there, right? So this is going to be causing what? This is going to be causing the canned food, right? So always remember, just keep the things in oxygen just after taking it out, right? This is how, just cook it properly and then you try to eat those canned food, right? Next thing we are having is going to be your Clostridium perfringens, which can cause generally food poisoning, generally happening with the reheated food, right? They are going to cause what? Your reheated food can cause this particular one, and this is going to activate what? This is going to activate vomiting in the person. The person will start to have what? Vomiting sensations will be there, and this person can have vomiting after eating the curries from the fridge, which was like, you know, you reheated it, but still some toxins are going to be there, and these toxins will produce their effect, right? Next one is your E. coli O157H7. This is an important one. This is also your PYQ. It is associated with the undercooked meat. If you are eating undercooked meat, right, this is going to activate your some toxins, right? This is going to be your enterohemorrhagic E. coli toxin is going to be there, which is has got some association with O157H7, and it can lead to hemorrhagic uremic syndrome. You can also have hemorrhagic colitis can also happen, right? So this is going to be happening due to undercooked meat. Remember, I told you undercooked meat, undercooked fish can lead to what? Even it can, undercooked fish can cause what? Your cancers, right? It can cause cancers in your body, right? So these things are going to be there. What about this salmonella? It is going to cause what kind of diarrhea, and what is going to be the route of administration is going to be what? Fecal-oral route of administration, generally happening with ingesting the dirty water, right? And poultry products are also important for this particular one. Poultry products, your eggs are also important, right? Even this thing can enter into the intact egg. So those people who are eating the eggs directly, they are consuming the eggs directly into the milk. It is not recommended. You need to cook it well and then you should be having it, right? Because the salmonella can also penetrate the intact egg, right? So agar कच्चा अंडा खाओगे तो this thing can enter into your body, right? Poultry products can also have this particular one. And what kind of diarrhea this person will be having? What kind of diarrhea? Yes, very good. This is going to have what? Pea soup kind of diarrhea. Pea soup diarrhea can be there, right? Vibrio parahemolyticus. Next one is going to be Vibrio parahemolyticus, going to cause what kind of poisoning? Seafood poisoning. Seafood poisoning. Someone is going to go and suddenly start to have what? Bouts of vomiting and diarrhea after 16 to 24 hours, right? What is the incubation period? Like the person is eating oysters or something, maybe, I don't know, right? The person is eating some seafood, and post eating the seafood, this person will start to have what? After 16 to 24 hours, this person will start to have what? Symptoms of this particular one, food poisoning. This is going to be due to what? Vibrio parahemolyticus. Vibrio parahemolyticus, right? So these are going to be some food poisonings they can write you. So what are the symptoms they can write you in the food poisoning cases? They will be always giving you history, how to read the question. There will be a long stem question. You need to think about the first thing, history. You need to look at something is eaten up by the person, right? The person is eating something, the person has got like Chinese fried rice can be there, meat products can be there, milk product can be there, canned food can be there, all these things can be there, your seafood can be there. So all these food products are going to be there, and post eating the food products, you need to calculate, you need to just think about it, the incubation period, and then you need to think about the symptoms. What are the symptoms? Whether the person is having vomiting or whether the person is having diarrhea sorry, along with the fever. If the fever is present, this is going to be a bacterial cause, right? If fever is absent, it can be due to amoebic cause also, right? It can be due to viral also, right? So this is going to be how you are making your diagnosis, and you will reach your answers, right? So this is going to be what? The answers, right? You should avoid everything at seafood one day before visit to Goa. I think you should be studying right now, should not do the visits, right? And make sure the last important few days are left, right? This is the last week that you are left with, right? Remember, you need to take care of your health at the utmost, right? This is the most important thing that can be, like, this is my experience. You know, people will get ill just 2 days or one day before the examination. You have to keep this thing in your mind. There might be mosquitoes, there might be some food that can infect you. Make sure you will not enter into this particular trap. Try to eat healthy, as healthy as can, as healthy as you can. Try to eat as healthy as you can. Try to protect yourself for the next 7 days, because this is going to be just a simple mistake. You eat outside some pani puri or something, right? And that can lead to infection, and that infection can lead to destructions. You understand this particular thing, right? So make sure you will have good food for the next few days and take care of your health. This is the advice that I can give you, right? Because I know people who had diarrhea in the day of examination, or vomiting, because they are traveling here and there, and due to travel, you know, they eat parathas or something, chole bhature outside, and that would cause troubles in the examination, right? So make sure you pick good things, even if it is costly. Just try to have something good, right? So this is going to be the thing. Now about the UTI, right? What are the symptoms of UTI that can be given in the examination, and how you will identify this UTI? So for UTI, the symptoms are going to be what? They can write you, the patient is having what? Burning micturition is going to be there, right? Patient is having increase in frequency of urination. Patient might have what? Fever. Fever can be plus minus, right? So these are going to be what? These are going to be burning micturition, increase in frequency, painful micturition is going to be there. So these are going to be the symptoms the patient is coming up with, right? Patient will be having pain in the lower abdominal area, and these symptoms are going to be written in the questions, right? So you need to identify this is a case of what? This is a case of urinary tract infection. And once you get to this urinary tract infection, you think about second thing, what is the most common cause for UTI? So what is going to be the most common cause for UTI? The answer will be most common cause for UTI. अगर वो पूछते हैं this patient is coming to you with these symptoms, the answer is going to be what? Most common cause for UTI is going to be what? This is going to be E. coli, right? And for E. coli, you can use what? Simple drugs. You can use Norflox, nitrofurantoin. This is going to kill this particular E. coli infection, and you will be fine, right? So this is going to be what? UTI. But also remember one more question they are asking. In fact, they asked this question in the last examination. Patient is on certain drug and he start to have what? Urinary tract infection. So what is that drug that can lead to increase in glucose release in the urine, right? So the patient is a diabetic patient. They have written, in the question they have written what? The glucose. They have written the HbA1c. HbA1c was high, the glucose was high, and patient was prescribed some drug, right? What was the drug the patient was prescribed? These are your gliflozins, right? And this gliflozin has got a specific side effect of what? UTI. This gliflozins, glucose flows in urine, so that means they will block the SGLT2, and now what will happen? Extra glucose will go out into the urine. When the urine is having that particular extra glucose, bacteria will come to do the party, right? And this bacteria will come and the person will start to have what? UTI. So what is the definitive management in this particular case? Try to stop these gliflozins, because the more glucose will come, the more bacteria will come. Even if you take antibiotic in this particular case, what will happen? The glucose will attract some more bacteria. Maybe this time E. coli, next time maybe Staph aureus, maybe next time Saprophyticus might come, right? So the most important thing, if person is prone to UTI, we are going to change the drugs, right? So this is going to be the thing. Yes, dapagliflozin, empagliflozin, right? This is going to be what? E. coli, right? Staph saprophyticus is going to be the another question they have asked you. Remember, if they have written somewhere in the question gram positive cocci, or maybe they can write you young sexually active female. If this is written in the question, you have to mark your answer Staph saprophyticus. Staph saprophyticus. Another question they are asking about the Staph saprophyticus. I told you, do not take stress. No stress is the mnemonic, right? So what is this? What is this? It is going to be novobiocin resistant. Is going to be which one? Staph saprophyticus. The Staph saprophyticus is resistant to novobiocin, right? And Staph epidermidis is sensitive to novobiocin. So these two things you have to keep in mind. So question number 1, most common cause of UTI, answer will be E. coli. Most common gram positive organism causing UTI, the answer will be Staph saprophyticus. Staph saprophyticus, right? If they write you there are large mucoid colonies coming in the urine, large mucoid colonies coming in the urine, then you need to think about what? Klebsiella, right? Then you need to think about Klebsiella. In the lungs, this Klebsiella can cause pneumonia. And what pneumonia? This pneumonia is known as what? This pneumonia will be having a special bulging fissure sign will be there, red currant jelly sputum will be there, right? So that is going to be what? That is going to be your Klebsiella. That is going to be your Klebsiella, right? In the another one, we are having what? We are having large mucoid colonies coming from the urine. This is going to be again Klebsiella, right? This enterococcus infection you can get after the hospital admission, right? And after hospital admission, what we call it, this particular one, we are going to call it as a nosocomial infection, right? So this is going to be the most common nosocomial infection that you are going to get. And remember, this enterococcus is also drug resistant because it is present in the hospital. It has got vancomycin resistant also. We can have VRE, vancomycin resistant enterococci can be there. So it is going to be having high resistance is going to be there. High drug resistant is there. And this high drug resistant is again due to what? Again due to transduction. Again due to transduction. The next one we are having is protease. This protease is going to cause what? It is going to produce an enzyme. This is going to be a urease positive organism, right? So it will convert your urea into ammonia. It will convert your urea into ammonia. And this ammonia will alter the pH, right? This will alter the pH leading to what? Stone formation. What stone? What stone is going to be there? This is going to cause what? Struvite stone formation. So this protease is going to be a urease positive organism which can alter the pH, and that would lead to what? Again, UTI along with the stone formation. Okay. Next one is going to be Pseudomonas aeruginosa. This is going to be associated with the catheter beta in the immunocompromised patient. Remember this Pseudomonas. I told you this Pseudomonas is a nasty bacteria which grows in the disinfectant, right? All the bacteria are dead due to disinfectant. This bacteria grows in the disinfectant, first thing. Second thing about this Pseudomonas, it is going to kill the burn wound patient, right? Burn wound patient, burn unit. Remember, you will enter into burn unit. This is going to be the Pseudomonas which will produce the pigments, green, yellow color pigments are going to be there. Pyocyanin, pyoverdin, and this bacteria is the killer for those people, right? वो जो burn वाले लोग मर जाते हैं, क्यों मर जाते हैं? Because of this Pseudomonas. Sir, इससे बचना कैसे? How we are going to save our patient? You need to do the dressing, right? You tell me what is the compound we are going to use for the dressing. Yes. Very good. One more important point you are mentioning, Shubham, right? This is going to be a long-term catheter insertion, right? So this is a catheter. Long-term catheter can lead to what? Again, Pseudomonas aeruginosa infection. Person is taking antibiotics, you are giving ceftriaxone, you are giving amoxicillin, you are giving azithromycin to the patient, but patient is not getting recovered. Patient का बुखार ठीक नहीं हो रहा, right? The patient is a catheterized or a bedridden patient. आपने सारी दवाइयाँ try कर ली जो आप कर सकते हो basically, right? And the person is not getting corrected. You go for a culture and you will find out ultimately the patient is having what? Pseudomonas aeruginosa, and now you need to go for higher antibiotics like meropenem, right? And this is how this person is going to get corrected, right? तो वो दवाई देने के बाद में patient ठीक होगा, otherwise this fever is not going to go away with the simple thing, right? So whenever you will find troubles, right, what will happen? This is going to be what? Pseudomonas aeruginosa generally, right? Generally happening in immunocompromised patients, right? Catheter induced patients, your HIV patients, your cancer patients, right? So these are going to be the one, right? And they are highly drug resistant. Normal drugs are not working here. And this is a aerobic bacteria beta. Pseudomonas is a aerobic bacteria. I told you aerobic bacteria कहाँ होगा? जहाँ ऑक्सीजन होगा. ऑक्सीजन कहाँ होता है? या तो स्किन पे होता है या लंग्स में होता है बहुत सारा ऑक्सीजन. So this is going to cause what? This is important cause for ventilator associated pneumonia also, right? Those people who are on ventilators, they can get this Pseudomonas aeruginosa infection, right? So these are some important point about this Pseudomonas aeruginosa, right? You can identify the cases of UTI by looking at these symptoms, and you can identify with the keywords. Micro is about keywords. It is going to be a subject where you can score 100% marks. 100% strike rate can be there if you read your notes well. There are going to be at least 19 to 20 क्वेश्चन you can appreciate, and these questions are going to be totally doable, right? सारे doable question होंगे. You just need to look for the keywords that we have learned through like our studies. पिछले 5 महीने में हमने यही सीखा है, how to approach to the questions, right? How to read the keywords, how to identify patient is having this, that, what is the age, what is the thing we are going to look for, है ना? हम लोगों ने इन सब चीज़ों में यही सीखा है, right? So this is what we are going to apply. These are the keywords that you need to remember, right? The next thing we are having is about the E. coli. What is this E. coli, right? E. coli is going to be the bacteria which is going to be most common cause for UTI, we already know, but it is an important cause for what? Diarrhea also. It is an important cause for diarrhea. And what are the strains which can cause diarrhea? These are going to be your Pitta, right? What are the Pitta? EPEC, that is going to be enteropathogenic E. coli. Enteropathogenic E. coli. EIEC is entero-invasive E. coli. ETEC is enterotoxigenic E. coli. EHEC is enterohemorrhagic E. coli. And EAEC is going to be enteroaggregative E. coli. Right? So these are the names of these particular E. coli, and these are the 5 different strains of E. coli which can cause what? Diarrhea in different age group patient, right? So just try to remember, P for pathogenic, it is going to cause what? Diarrhea in the pediatric age group patient. P for pathogenic, P for pediatric age group patient. In the INI set, they have asked one question about the lesions, and it will be having attaching and effacing lesions, right? Image you can see in the workbooks, right? So this person will be having what? Attachment and effacement type of lesions. This is going to be due to EPEC. एंटेरो इन्वेसिव ई कोलाई, इन्वेजन मतलब क्या हो गया, ब्लड वेसल के अंदर घुसेगा, राइट। अगर किसी को नॉर्मल डायरिया इज गोइंग टू बी व्हाट, डायरिया मींस लूज स्टूल इज कमिंग more than 3 times, right? More than 3 times if the person is going to have what, loose stool coming, यानी पानी वाला स्टूल आ रहा है, this is known as what, डायरिया। अगर उस डायरिया के साथ में म्यूकस प्लस योर ब्लड स्टेन्स आर आल्सो कमिंग, राइट, दिस इज गोइंग टू बिकम व्हाट, डिसेंट्री। एंड इन्वेजन मींस ये ब्लड वेसल तक जाएगा तो कहीं ना कहीं डैमेज करेगा, इट कैन लीड टू व्हाट, डिसेंट्री। So entero-invasive E. coli is going to cause what, dysentery, bloody diarrhea. There are only few causes of bloody diarrhea that we need to remember. We'll discuss about them, right? ETEC is going to be what, enterotoxigenic E. coli, T for toxigenic, T for traveler's diarrhea. It is going to cause what, traveler's diarrhea, right? It is going to cause what, traveler's diarrhea. Enterohemorrhagic, I told you it has got association with, in your pathology also you have studied about this thing, O157H7. The strains are going to be there, O157H7, it has got association with hemorrhagic colitis, hemorrhagic uremic syndrome. So this is important, enterohemorrhagic E. coli. It is also going to cause what, dysentery. Hemorrhagic means bleeding will be there, it can also lead to what, dysentery. So till now 3 causes you can remember about dysentery. One is Campylobacter jejuni, I told you. Second one is going to be what, Campylobacter jejuni due to uncooked food, right? It has got a temperature 42 degree, like, right? So this is going to be the thing. Enteroaggregating is going to cause what, persistent type of diarrhea. It is going to cause persistent type of diarrhea. Persistent type of diarrhea will be there, right? So these are going to be the 5 important strains that you need to remember. T for traveler's diarrhea, P for pathogenic, P for pediatric, invasive is dysentery, and this O157H7 is very, very important. If they ask you most common cause for diarrhea, the answer will be what, beta? The answer will be rotavirus. Sir, ये क्या बोल रहे हो, right? Rotavirus, yes, the answer will be rotavirus. It is going to be the most common diarrhea is due to what, virus, right? But if they ask you most common cause for bacterial diarrhea, this is going to become what? Now you can go for the E. coli, now you can go for the other bacteria, right? What about the culture medias, right? In the adults, in the childhood, in the children's, rotavirus. In the adults, you can remember norovirus, right? Norovirus. Culture medias are going to be what? For this E. coli, the culture media is going to be what? Clade. We can also use what? McConkey. Clade is cysteine lactose electrolyte deficient media, whereas this is going to be, another one is going to be what? McConkey. This E. coli can cause what? Meningitis and neonatal meningitis. Neonate means एक बच्चा जो है 28 दिन तक का, 0 to 28 days is going to be what? A neonate. 0 to 7 is early neonate, 7 to 28 is a late neonate, and this is the most common cause for neonatal meningitis. If they ask you, in a neonate, if the person is going to have inflammation of the meninges like dura mater, arachnoid and pia mater, this is going to be known as neonatal meningitis, right? And the most common cause, sir हम ये most common cause क्यों पढ़ते बार बार? Because we need to give our empirical treatment. बेटा empirical treatment मतलब हमको हमारे दिमाग से एक दवाई देनी है. When the patient is going to come, we are not going to see any reports. हम बिना रिपोर्ट के एक दवाई तो स्टार्ट करेंगे, पेशेंट में कुछ ट्रीटमेंट तो देंगे, राइट? सो दिस ट्रीटमेंट इज बेस्ड ऑन दैट मोस्ट कॉमन कॉज, राइट? सो most common cause for this particular one is going to be what? GBS. This is going to be group B streptococci, right? This is going to be Streptococcus agalacti, followed by E. coli. Most common cause is followed by E. coli, followed by Klebsiella. So these 3 important cause you can remember, that is going to be group B streptococcus, you can have E. coli, you can have what? Klebsiella, right? We need to start the treatment first, right? So just thinking these things in mind, if adult patient is coming, I start what? Ceftriaxone, vancomycin, right? Why I'm starting that particular thing? Because I know about this particular one, right? This most common cause of pneumonia in adult patient is streptococcus pneumonia. Most common cause of meningitis in an adult patient is going to be what? A streptococcus pneumonia. So I know about the cause. If you know the most important, most common cause, you can give the first diagnosis, first treatment, that is known as empirical treatment, okay? So this is going to be the types of strains that we are having for the E. coli, most common cause for UTI as well as your diarrhea beta, right? Next thing, next thing we are having is Treponema pallidum, very important and exam favorite. This is going to be what? Syphilis that we are having. What kind of microscopy we have seen? We have seen the dark ground microscopy in the chapter number, in the slide number 1. Here you are having this dark ground and you are having this spirochete organism. So this is going to be what dark ground microscopy we are going to use for this Treponema pallidum. What is the motility for Treponema pallidum? Try to answer, right? Corkscrew motility is going to be there, right? We are having syphilis. How many types of syphilis? We can remember the first one is going to be what? Primary syphilis. The primary syphilis is known as what? Chancre. We are having this chancre. This is going to be what? Single painless genital ulcer. So this is going to be a single painless genital ulcer. This is known as what? Primary syphilis. And secondary syphilis, you will be having what? Condyloma lata. What is this condyloma lata? Lata had syphilis, remember, right? So this lata will be having what? The vascular lesions on the palms and soles. If you find this picture, palm and sole picture, think about what? Secondary syphilis. Along with that, you can also have from derma, you can remember, in secondary syphilis, you will also find what? Mouth eaten alopecia, beta. Mouth eaten alopecia, right? And if you will find another picture in the examination which is having hand, foot and mouth picture is also given, this is going to become what? अगर ये तीन फोटो साथ में आ रही है, hand, foot और mouth, what is going to be the answer? This is going to become what? HFMD, hand, foot, mouth disease, which I will teach you further, right? So this is these two pictures are given. This is going to go in the favor of what? Secondary syphilis. So we have got our primary syphilis, secondary syphilis. We are having latent syphilis. Latent will be having early latent and late latent forms, and then we have got the tertiary syphilis, which is going to have what? Granuloma formation. Long term thing will lead to what? Granuloma, and these granulomas are also known as gamma, right? So we can have this gamma formation, and this is the gamma that you can appreciate. This is the gamma. This is going to be what? Gamma formation. We can also have what? Neurosyphilis. You can have Tape's dorsalis. These are the complications. We can have congenital syphilis also. In congenital, you will be having what? Saddle nose and Hutchinson teeth is going to be there, right? The Hutchinson teeth is going to be there. This is what you read in the pediatrics, right? So this is going to be the different types of syphilis that we are having. What is the drug of choice for syphilis? The drug of choice is going to be what? Benzathine penicillin G, right? So this is going to be the drug of choice that we are going to use for the syphilis, right? Long-term drug we need to give prophylaxis and treatment for this particular one, right? So this is going to be the syphilis we are having: primary, secondary, early latent, late latent, tertiary, and neurosyphilis and congenital syphilis. These are the types of syphilis you have to remember, right? Whenever they will write you a case, truck driver, remember the truck driver question they are asking very commonly. A truck driver is coming to you with single painless genital ulcer. You need to think about what? This is going to go in the favor of syphilis, right? This is going to go in the favor of syphilis. Another, this particular person, who is this, and try to answer what is the appearance it is trying to tell me, right? Yes, hand, foot, mouth disease is due to Coxsackie A16, right? So this is going to be a case of what? This is a case of, this is an image which is telling us about the bioterrorism agent that we are having. This is known as what? Bacillus anthracis. Bacillus anthracis. Bacillus anthracis is a which category bioterrorism agent? The answer will be category A bioterrorism agent, right? It is going to have the spores, and these spores are going to be motile or non-motile? They are going to be non-motile spores are going to be there, right? How many types of anthracis we can have? How many types of anthracis we can have? We can have cutaneous anthracis, that is cutaneous anthracis. We can have intestinal anthracis, and we can have pulmonary anthracis. So we can have pulmonary anthracis and we can have intestinal anthracis. Cutaneous anthracis, pulmonary anthracis, and intestinal anthracis. And this anthrax is going to be important cause for what? It is a category A bioterrorism agent, right? So what happens? These are the people, they have left the spores, right? They have segregated all the spores, and then they are going to spread these spores to the areas. And now what will happen? The spores will come in contact with the person, right? So once the spore will come in contact with the person, he will start to have what? Cutaneous anthracis, right? And this cutaneous anthracis is generally commonly seen with other people also, those people who are going to carry the skin of the dead animals, right? So this these people are known as what? Hide porters, right? Hide means dead skin, right? So those people who are going to take care or port the dead skin, they can have what? They can have a disease, and this person is going to have what? A pustule, which is going to turn black, right? And which is going to turn into a black color scar. So this is known as what? Malignant pustule is going to be there. So first it will start to have a pustule, and then it will turn into a malignant, not malignant. Malignant means non-cancerous, but it is a dangerous thing, right? So this is going to be the person who is going to have a black color scar will be there. Black scar will be there. This is known as what? This is known as Hyde-Porter's disease. This is also known as cutaneous anthracis. Two important things you have to remember in the examination. In the last exam, they have asked you one question about what? Black color lesion in farmer, black color lesion person going to the forest. You just need to remember two important thing. Black color lesion, first one is going to be, in this particular one, person going to the forest will be having what? Black color painless lesion. This is going to be what? This is your malignant pustule. This is the case of what? Anthracis. One important thing is going to be what? Anthracis. Second important black color lesion on the body is going to be seen with what? This is seen in the scrub typhus, right? In the scrub typhus, you can also have black color scar at the site of the bite mark. So you need to identify the bite mark to identify the scrub typhus. Scrub typhus के लिए है ना. तो ये दो चीजें याद रखनी है. फॉरेस्ट में दो ही black lesions are going to be there. Two important black lesion. One is going to be anthracis. Another one is going to be your scrub typhus. Right? These two important entities you can remember. The next one we are having is about the pulmonary anthracis. When the person is going to inhale those pores will come into the lungs. Once they will come into the lungs, they will cause what? Mediastinal hemorrhagic necrosis, right? They are going to cause what? Mediastinal hemorrhagic necrosis will be there, right? And this thing can lead to death of the person, right? So this is going to be what? Hemorrhagic necrosis can happen, right? And this particular thing will happen commonly with the people who are sorting the wools. Those people who are sorting the wools. So that is why this disease is also known as wool sorters disease. Right? And this wool sorters disease, we are going to pre-treat this wool, right? Earlier this thing was happening, but nowadays we are pre-treating the wool with formaldehyde. So to avoid this particular thing, we are going to do what? We are going to pre-treat the wool with formaldehyde. And this process is known as, this is your exam question, we are pre-treating the wool with formaldehyde. This process is known as what? This process is known as duckering. This process is known as duckering, right? So the important thing is going to be what? What is duckering? Pre-treating the wool with formaldehyde. What is this? Malignant pustule is seen in Hyde-Porter's disease or cutaneous anthracis. What is the drug of choice you can use? You can use ciprofloxacin, right? What is the appearance you are going to get? That is also important. This is going to be your Miss Bamboo's appearance, right? So what is this Miss Bamboo appearance you are going to get in the anthracis? You can remember M for Medusa head appearance. So you can appreciate this is the lady which is having what? This Medusa head appearance is going to be there. Multiple snakes like things coming out. I for inverted fir tree. You can remember this is the inverted fir tree we are having. S for string of pearl. मोतियों की माला पहन रखी, right? This is going to be Miss Bamboo, means she is having bamboo on the one hand. This is going to be bamboo stick appearance. And what is the culture media? This is going to be the plate. दूसरे हाथ में क्या है मैडम के? This is going to be the plate. So you can remember this is going to be what? Miss Bamboo appearance, and this plate is the culture media. And toxins also you can remember, she is going to go with this particular attire. She is going to watch the England Premier League. So these are going to be the toxins we can have. Edema factor, protective factor, and lethal factors are going to be the toxins. So you can remember Miss Bamboo. It is going to be easy for you to remember, right? What is Miss Bamboo? There is a miss. She is having what? Medusa head appearance. I for inverted fir tree appearance is going to be there. S for string of pearl appearance is going to be there. One hand she is going to have what? Bamboo. Another hand she is going to have the plate, right? What kind of toxins in the plate? She will be having EPL. That is going to be edema factor, protective factor, lethal factor, and the drug of choice is ciprofloxacin. Everything is done, right? So this is about the Bacillus anthracis, important question. This is a one more important thing, all the capsulated organisms will be having capsules made up of what? Polysaccharide, but this particular one is going to have capsule made up of polypeptide. So this particular one will be having capsules made up of polypeptide, right? So this is going to be what? Bacillus anthracis. This is going to be what? Bacillus anthracis. Now we are having what? Another case. This is going to be the case of meningitis. So how you will identify meningitis? What are the symptoms of meningitis? So to diagnose meningitis will be a clinical diagnosis, right? So this is going to be what? We are going to have certain meninges. What are the meninges we are having? They tell me. We are having dura mater, we are having arachnoid mater, we are having pia mater. This brain is a closed cavity. We enter into the brain, you will find the skin, you will be having scalp, right? You will be having skull. So skull is a closed cavity. Yes or no? Skull is a closed cavity. Then you have dura mater, arachnoid mater, pia mater, right? And below this pia mater will be brain parenchyma, right? Brain parenchyma, which will be having those sulci and gyri are going to be there, and they do not have any space, right? So here you will be having those sulci and gyri will be there, gray matter, white matter will be there, and these three layers are going to be there. These are going to be what? Dura mater, arachnoid and pia mater, right? Just giving you a quick recap for both the things. You can also remember meningitis and other thing also, right? So remember, above dura mater will be there a space. This space is known as epidural space, and here you can have what? EDH. If the blood is going to get accumulated here in this space, this is known as EDH. If you are having blood accumulation in this particular area below the dura mater, this is going to be known as subdural hemorrhage, right? Subdural hematoma. Below the arachnoid you are also having some space, and if something is going to be there, this is known as subarachnoid hemorrhage. So three hemorrhage you can remember, three areas you can have blood accumulation. You can have EDH, which is going to give you your idli. This is going to give you what? This is going to give you your crescent shape appearance, right? And on the radiology, and this is going to have what? You can look for the circle of Willis we are having, right? This is going to be what? Subarachnoid hemorrhage characterized by thunderclap headache, right? So this is going to be the basic thing. If you are having inflammation of these three layers, this is known as what? This is known as meningitis, beta. These three layers are known as meninges, and this is known as meningitis. Whenever you are having inflammation of brain parenchyma, your sulci, gyri, your gray matter, white matter, this is known as what? This is known as encephalitis. This is known as encephalitis, right? And below these layers you will be having your hindbrain, and below hindbrain you will be having your spinal cord. If you are having problem with the spinal cord, if you are having problem or issues with the spinal cord, this is going to become what? This is going to become what? Myelitis. इसको क्या बोलते हैं बेटा? इसको बोलते हैं myelitis. So you can have transverse myelitis, you can have poliomyelitis, right? So myelitis means problem with the spinal cord, encephalitis means problem with the brain parenchyma, meningitis means problem with the meninges. Three meninges sorted, right? दिमाग में sorted होना चाहिए, right? Next thing, encephalitis में क्या symptom आता है बेटा? The symptom they will be writing you in the question is going to be what? Seizures are going to be there, है ना? एपिलेप्सी कब बोलते हैं? जब कोई कॉज नहीं होता तब एपिलेप्सी, देन वी कॉल इट एज अ एपिलेप्सी. इफ द कॉज इज प्रेजेंट वी कॉल इट एज अ सीजर. सो यू आर गोइंग टू हैव व्हाट? एनकेफेलाइटिस will present with seizures, right? Anytime you are having touching of these things, you can have what? Seizures, right? It is going to be atypical kind of seizures, right? Whereas whenever you are having inflammation of these meninges, they are going to put some pressure, right? And these symptoms are going to be what? They can cause certain symptoms, like they will touch your CTZ center, which will lead to what? Vomiting, right? So you can have what? You can have in the question, they can write you patient is having headache, patient is having photophobia. Because of the pressure, it can also compress your optic nerve, right? You patient can have photophobia, patient can have what? Fever. These are going to be what? Symptoms, and some of them are going to be what? Signs are going to be there, signs that you can check as a doctor. Symptoms are going to be, patient will be telling you, right? Patient can have what? Nuchal rigidity, neck stiffness is going to be there, and then you check for certain signs, and these signs are going to be what? Positive, right? Patient can have what? Projectile vomiting, nausea, vomiting, fever. Vomiting can be there in the patient, right? So these are going to be the common symptoms, right? What signs they can write you positive? This is going to be Kernick sign positive, your Brudzinski sign positive. So these signs can come out to be positive. This is going to be Kernick positive, Brudzinski positive. This is going to be a case of what? If you find these symptoms, patient is having elevated temperature, all these things. Along with that, nowadays these are the earlier identification. Nowadays they are also writing you these things, right? And sometimes they can also write you values. If they are asking questions from the medicine, they can write you values, right? We will try to identify by looking at CSF how you can identify whether what kind of meningitis this person is having, right? So these are going to be what symptoms of meningitis, right? Now let's talk about the most important causes for meningitis. What are the most important causes for meningitis? In a neonate, we already know, if they talk to you about a neonate, this is going to be group B streptococci, also known as agalacti, is the most common cause, followed by E. coli, followed by Klebsiella. So if a neonate is coming to you with these symptoms, you need to think about it is due to agalacti or E. coli or due to Klebsiella. These are the first line things that should be coming into your mind. But if an adult patient is going to come to you, this is going to be what? Streptococcus pneumonia is the most common cause. Streptococcus pneumonia or pneumococcus is the most common cause. One more thing I told you is about the elderly or elderly or children. Elderly or children, this can be due to what? It can be due to listeria. The important one is going to be what? Listeria. If they tell you about HIV positive patient coming to you with the symptoms of meningitis, this is going to be your cryptococcal meningitis. Very, very important, cryptococcal meningitis, capsulated fungus. Right, so this is how you can identify the causes for this meningitis, right? These are going to be the basic causes. Now for the diagnosis and treatment, you are going to have what? How to identify bacteria. Bacteria will eat up the glucose. Dekho, simple thing is, virus is coming into your body just for the replication, but bacteria is coming into your body. He has got the replicating things, he has got phyla, he has got other methods also replication, right? But the bacteria is coming into your body for the search of food, right? And what is the food that we are having? What is the food that we are having beta here? The food we are going to have is going to be what? The glucose, right? So what will happen when the bacteria is going to come into your body? They are going to eat up the glucose. So what will happen? This amount of glucose is going to get low, right? This is going to decrease the levels of glucose. In medicine, they can also write you the values, so you can refer to these values also. Sometimes they can write you the glucose is going to be what? 25. So you need to have a range. If they have not given you the range, generally they will give you, but in NEET PG they don't give you, right? So just remember these three values. If you can remember, that is going to be important, or at least these two you can remember. This is going to help you out. Okay, so first thing is going to be what happens to the glucose. Glucose will be very low. बैक्टीरिया अगर खाएगा तो कुछ ना कुछ निकालेगा भी. सिंपल अंडरस्टैंडिंग, दिस इज गोइंग टू take out some fluid, and this is going to be yellow or turbid fluid is going to be there. It is going to eat up and it is going to pass the stool, thereby leading to what? CSF discoloration. Normal CSF is going to be clear in color, right? Apart from that, this protein levels are also going to be very high, right? This is going to be high. And the first line antibody, first line neutrophils, first line WBCs which are going to come into the action are going to be what? Neutrophils. They are going to come into the action, so you will be having lots of neutrophils. We already know in bacterial infection the levels of neutrophils will be high, so you can have neutrophilia or polymorphonuclear cells are going to be there. So these things are written. You think about what? Bacterial meningitis, right? Or simple understanding, a patient is coming to me with all these symptoms. I'm going to take out the CSF for identification. When I'm going to take out the CSF, if the color is yellow or turbid, I will think about this patient might be having some bacterial infection. I start my treatment immediately. But if the CSF is clear and the person will be having problem, I think about what? There might be issues with the viral or some other kind of meningitis can be there, right? So in viral cases, what will happen? Virus is coming into your body just for replication, not for the not for the food, right? So what will happen? The glucose is going to become normal and the fluid is going to be clear. It is not going to, it is just going to divide, not going to eat up and pass something, right? But the important cells which are going to be there in the long term, in the earlier phase acute reactions, you are going to have what? Neutrophils coming. For long term or viral infections, the lymphocytes are going to be high, so here you will be having high levels of lymphocytes. What about this tubercular? Tubercular is going to be what? It is going to be a most common cause for meningitis. It is going to be important cause for hydrocephalus in the patient. The female patients or young patient or adult patients are coming to you with the symptoms of hydrocephalus. The most common cause for this hydrocephalus is going to be what? This tubercular meningitis. It is going to produce some proteins and all those things, they are going to clog the ventricles and leading to what accumulation of CSF in the brain, and that would lead to what hydrocephalus, and this hydrocephalus will produce pressure and the person will be having symptoms, right? So this is going to be having what, when you are going to take out the CSF, this is going to be turbid, and when you are going to start keep it standing for some time, you are going to get this is special feature, this is known as cobweb coagulum, right? You are going to find in the slide, I've shown you the picture in the notes. This is going to be the cobweb coagulum you are going to get on the base, right? What about the glucose? This is a bacterial infection. Glucose will be low, but it is a chronic infection. So you know what happens? The levels of lymphocytes will be high here, right? So you need to think about this particular one. Whenever they are asking, you need to give special focus. Cobweb coagulum, if you think about these two things are going to go in the favor of what? Bacterial one, but this is going to make you confused. So always remember, long-term infections can lead to lymphocyte increase, right? So all these things are going to be telling you about the diagnosis, and now if you know the diagnosis, you can give your treatment also, right? Viral general treatment is going to be what? Water. Tubercular, we are going to give what? Steroid also we can use to decrease the pressure of the brain, right? So some drugs like mannitol we can use acetazolamide for decreasing the pressure. First we need to decrease the pressure, then we start what? ADT, which is the definitive treatment. And in the cases of bacterial, you can use the drugs which are going to be according to the cause, right? According to the cause, right? Yes, CSF pressure is also going to be extremely high in this particular case. When we are going to enter into the space, this CSF is normally coming very slowly, right? With this limited drip by drip it is coming, but if this is a case, the CSF pressure will be high. We can appreciate this thing, right? The next important disease we are going to encounter in our examination, they are asking questions, is about the pneumonia, right? Meningitis identify कर लोगे, meningitis का question कर लोगे, right? The next thing we are having is about the pneumonia. What is going to be the symptoms of pneumonia? They are going to write you, right? Pneumo मतलब होता है pneumo means air, right? So what happens in pneumonia? How many types of pneumonias we can appreciate, and what are the causative agents behind them? So we need to think about all these things, and what are the symptoms? So in the questions they are going to write you, the patient is having cough, patient is having high respiratory rate is going to get increased, right? It will be baby अगर होगा तो उसका बहुत ज़्यादा respiration होगा, respiratory rate बढ़ जाएगा. Patient can have what? Fever. Patient is not able to breathe properly. Basically, you know what is happening? This is the alveoli. It is going to have accumulation of fluid. Normally, this alveoli and blood vessels are going to be there, which are going to be responsible for what? For the exchange of gases, right? But you know what is happening in this particular case? We are having some accumulation of fluid over here, and because of this accumulation of fluid, do you think these gases can exchange very nicely? No. If these gases cannot exchange very nicely, what will happen? Your brain will activate certain responses to increase the oxygenation in the body, because body is going to have hypoxia. So body क्या करेगा? आपका respiratory center in the pons and medulla will activate your apneustic center, leading to what? Leading to increase in the respiration, है ना? K plus बढ़ जाएगा, H plus बढ़ जाएगा, तो CO2 बढ़ जाएगा body के अंदर. So body will start doing the hyperventilation. So person will start to have what? Hyperventilation, right? So this is going to be what? This is going to be pneumonia. Patient can have cough, increase in respiratory rate can be there, fever can be there, all the symptoms are going to be there, pulmonary symptoms are going to be there. This is going to go in the favor of pneumonia. We can have two important types of pneumonia. Beta. First one is typical pneumonia, another one is atypical pneumonia. दो निमोनिया याद रखने. टिपिकल निमोनिया मतलब लोबार निमोनिया, लोब्स में निमोनिया हो गया. तो जब आप एक्सरे देखोगे कोई भी एक लोब आपकी इन्वॉल्व हो रखी है, दिस इज गोइंग टू बी टिपिकल लोबार निमोनिया. इसमें क्या होता है? इन दिस पर्टिकुलर वन द सिम्टम्स आर गोइंग टू बी व्हाट? द सिम्टम्स आर गोइंग टू बी मोर. सिम्टम्स ज़्यादा आएंगे और डिजीज जो होगा वो कम होगा. disease is going to be less, because ये exactly कहाँ होता है? In the alveoli, right? This thing is going to happen in the alveoli, leading to more problems, severe problems, right? Severe problems, severe symptoms, you can say. Problem is not severe. It is going to involve one lobe, right? This is going to be what? Lobar pneumonia, right? So this is going to be what? Symptoms are more, but the disease is less, right? What is the most common cause for typical or lobar pneumonia? The answer will be what? Pneumococcus or streptococcus pneumonia. Right, this is the most common cause for pneumonia that is going to be there, right? More symptoms, less disease, right? You go for HRCT scan, you will find only 5% of the lungs are damaged. Another one is your another entity is atypical pneumonia, also known as what? Walking pneumonia. इसी को walking pneumonia भी बोलते हैं. क्यों बोलते हैं? Person will be walking, right? घूम रहा है बंदा, during COVID-19 people are walking, people are sitting at home, they are like छोटा मोटा कफ है, खांसी है, कुछ भी नहीं है, सब ठीक है, right? And suddenly they will admitted to the hospital and they will start to have what? They will start to have what? They will be having severe hypoxia, needing oxygen, and if they are not going to get the oxygen, they are dying, right? So this is what is happening in the walking pneumonia. Walking pneumonia, the most common cause for walking pneumonia is going to be mycoplasma, right? Remember, this is the most common cause for walking pneumonia, right? Apart from that, you can also have what? This walking pneumonia due to Legionella. Some bacterial infections can also cause this COVID-19, right? So atypical pneumonia matlab kya hai? Symptoms are going to be less and disease is going to be more when this person is admitted to the hospital. पहले थोड़ा cough और cold था, ज़्यादा कुछ नहीं था, but this person will start to have development of what? Pneumonia. और अब fluid कहाँ इकट्ठा हो रहा है? In the interstitial space, right? So what can happen? What can happen in this particular case? जब ये fluid पूरे lungs में white out lungs दिखते हैं, इसमें एक्सरे में कैसे lungs दिखेंगे? ARDS की situation होगी, acute respiratory distress syndrome will be there. And acute क्यों बोलते हैं? Because within a short span of time this person will start to have respiratory distress and he will be having white out lungs. Normally lungs कैसे दिखने चाहिए थे? Black, एयर ब्लैक होती है, बट यहाँ पर क्या हो गया? फ्लूइड एक्यूमुलेशन हो गया, सो द पर्सन विल बी हैविंग व्हाइट आउट लंग्स आर गोइंग टू बी देयर. और जब आप इनका एक्सरे करा के आगे देखते हो, यू आर गोइंग टू फाइंड व्हाट? दिस पर्सन इज हैविंग सीवियर डिसीज. 50 से 60% लंग डैमेज हो गए, राइट? एंड ओनली फ्यू पार्ट ऑफ द लंग इज वर्किंग, राइट? सो दिस इज गोइंग टू बी नोन एज एटिपिकल निमोनिया, यानी पहले सिम्टम्स कम थे और अचानक से when you go for identification you are finding that the disease is going to be a lot, right? And this is the case where you are going to try to revive your patient. This is going to be known as what? Atypical pneumonia, right? अब examiner हमसे क्या question पूछता है? You know about two types of pneumonia, one is typical pneumonia, one is atypical pneumonia, right? Mycoplasma में remember important question, mycoplasma do not have cell wall. अगर इसमें सेल वॉल नहीं है तो क्या सेल वॉल सिंथेसिस इनहिबिटर काम करेगी? नहीं करेगी. व्हाट आर द सेल वॉल सिंथेसिस इनहिबिटर्स? अगर किसी को माइकोप्लाज्मा निमोनिया हो गया तो क्या मैं एमोक्सिक्लेव खा सकता हूँ? क्या मैं सेप्ट्रेक्सोन लगवा सकता हूँ? or maybe I can shift to the meropenem, higher class antibiotic. No sir, nothing is working. Even peptides is not working, right? Why? Because all those drugs that I'm talking about are cell wall synthesis inhibitors, right? And this bacteria do not even have cell wall. Mycoplasma don't have cell wall, right? So that is the reason the drug of choice for mycoplasma infection is going to be what? What is the drug we are using? This is the reason why we are using what? Azithromycin, right? So this azithromycin is a protein synthesis inhibitor which is going to be working here. It is a macrolide that is going to be the drug we are going to use here, and that is the reason why we are having sore throat every time we take up azithromycin. Why we are not taking amoxicillin? आपको लगता है आपको हो गया atypical pneumonia, right? Or doctor be precautionary, they are starting this azithromycin, but this has got nothing to do with the bacteria because this is a static drug beta, right? Amoxicillin drugs, cefixime, ceftriaxone, all these things are sidal drugs, है ना? फिर भी हम azithromycin क्यों खा रहे हैं? क्योंकि हमको दे दी थी तो हम खा रहे हैं, right? So this is what is the basic funda that you also need to remember because you are going to pass within some time and you are going to go for your internship. So I just want you guys to become a good doctor, better doctor, right? So next thing we are having is about alcoholic patient, right? Whenever they will ask you a question about alcoholic patient coming to you with the symptoms of pneumonia, fever, headache, your respiratory distress might be there, your coughing is going to be there for such a long time, right? So in this particular case, you can mark your answer as Klebsiella. Klebsiella is going to be the patient with alcoholic patient with the symptoms of pneumonia. Here you will be having what? Two important entities. Red currant jelly sputum will be there. Red currant jelly sputum. Where do you find red currant jelly stool, beta? Red currant jelly stool कहाँ मिला था? Right, red currant jelly sputum along with bulging fissure sign. Along with bulging fissure sign. So this is going to go in the favor of what? Klebsiella pneumonia, right? Atypical pneumonia. Just now, just now we have seen mycoplasma, chlamydia, legionella, very important, central AC, legionella. Two things you have to remember in the question. Look for two keywords. Central AC अगर दिख रहा है और साथ में डर्टी वाटर अगर दिख रहा है, सेंट्रल एसी कोई कॉर्पोरेट कंपनी में बंदा काम कर रहा है और निमोनिया हो गया, दिस इज गोइंग टू बी योर लीजियोनेला, राइट? Yes, obviously, very good, right? This is going to be what? Red currant jelly stool in the N2 susception. And in fact, you can also remember this set of information. This azithromycin is a drug of choice in 4 cases. These are going to be your What are the 4 cases where the drug of choice is azithromycin? C for chancroid. In chancroid, the drug of choice is azithromycin, which is due to Haemophilus ducreyi. L for Legionella beta, which is also atypical type of pneumonia. A for atypical pneumonia. Atypical pneumonia, it is the drug of choice. And P for pertussis. Pertussis, right? Pertussis may be drug of choice क्या है? Azithromycin. So this clap you can remember, right? For azithromycin, right? All right, the next thing we are having, if a patient is having cystic fibrosis, the most common cause is going to be what? Pseudomonas. ये keywords मैं जो बता रहा हूँ ये याद रखने, ये एग्जाम में आते हैं. अगर पेशेंट को cystic fibrosis है, you need to mark the answer pseudomonas. Most common cause is pseudomonas, right? COPD patient coming to you with the symptoms of pneumonia, this is going to be either streptococcus pneumonia or influenza. ये जरूरी है क्या? क्या ये इंपॉर्टेंट है? पढ़ना इंपॉर्टेंट है क्या? एग्जामिनर पूछता है. यस, डेफिनेटली एग्जामिनर इज आल्सो आस्किंग, एंड दीज थिंग्स यू आर गोइंग टू एनकाउंटर ईच एंड एवरी डे. है ना? COPD is a very common thing. Lots of people are smoking nowadays. You know what happens after 40-50 years? They are going to get the COPD, and lots of people already having COPDs, right? They are coming to your hospital. The first important cause is going to be what? Pneumonia. Second one is going to be what? Influenza. Influenza is little bit lower because we are having in our national immunization schedule HIV, Haemophilus influenza B vaccination we are having, right? So that is why the most common cause remains what? Pneumonia, right? And then we start with ceftriaxone vancomycin, that we have to start, right? HIV positive patient coming to you with the symptoms of pneumonia, very very important question, multiple times repeated, pneumocystis cerevisiae pneumonia. CD4 count अगर 200 के आसपास दिख रहा है बेटा, CD4 count is around 200, definitely you need to mark what? This is going to be pneumocystis cerevisiae pneumonia. वेंटिलेटर एसोसिएटेड निमोनिया का मोस्ट कॉमन कॉज सूडोमोनास अगर ऑप्शन में नहीं है तो यू नीड टू मार्क व्हाट? यू नीड टू मार्क एसिनेटोबैक्टर. वेरी वेरी इंपॉर्टेंट. पीडियाट्रिक्स में आपको एसिनेटोबैक्टर ज्यादा मिलेगा. पीडियाट्रिक बुक इज हैविंग व्हाट? इंटरनेशनली नॉर्मल अंडरस्टैंडिंग इज व्हाट? इंटरनेशनली ये ज्यादा होता है एज कम्पेयर टू दिस पर्टिकुलर वन, राइट? इंडिया में ये ज्यादा होता है, राइट? सो डिपेंडिंग ऑन व्हाट दे आर आस्किंग, एक ही ऑप्शन में मिलेगा. यू नीड टू आइदर मार्क इट एसिनेटोबैक्टर और सूडोमोनास, राइट? Nosocomial infection, most common nosocomial infection is going to become what? Your staph aureus. And staph के बाद भी क्या होता है? MRSA, वर्सा, those infections can be there, right? So अगर किसी को pneumonia हो गया due to nosocomial, this is going to be MRSA और वर्सा pneumonia, right? This is going to be what? Pneumonia. अब CD4 count की बात कर लेते हैं. If in the question it is going to be written what? CD4 count is written somewhat around 500. है ना? कि HIV positive patient coming to you with the symptoms of HIV immunocompromised patient है. TB is the most common opportunistic infection that can happen at a level of CD4 count 500. CD4 count अगर 200 है, this is going to be plasma cell pneumonia और pneumocystis cerevisiae pneumonia, right? Last time image दिया था, this is the image of ping pong ball, crushed ping pong ball appearance, right? If the CD4 count is below 100, this is going to go in the favor of toxoplasma. Toxoplasma due to cat, right? So this is going to be toxoplasmosis can happen, right? And lastly we can have what? CD4 count अगर 50 दे रखा है, always remember cytomegalovirus, which is going to have what? Owli inclusion bodies. Owli inclusions is going to be there. Owli inclusions is going to be there, right? So this is going to be what? This is going to be the thing we are having. CD4 counts are important, right? Or also let me mention some important points. They can ask you some questions about HIV. So HIV positive patient coming to you with the symptoms of meningitis. If they will ask you about the symptoms of meningitis, you need to mark your answer cryptococcus meningitis. Cryptococcus meningitis. If they ask you HIV positive patient having pneumonia, the answer will be what? The answer will be plasma cell pneumonia or pneumocystis cerevisiae pneumonia, same thing. CD4 count यहाँ 200 होता है, right? If they ask you HIV positive patient coming to you with the symptoms of diarrhea, then you need to mark your answer as what? Coccidian parasites like Cryptosporidium parvum, Isospora, Cyclospora. Cryptosporidium, right? This is going to be the most important cause for diarrhea in HIV positive patient. HIV positive patient, most common opportunistic infection is going to be what? Tuberculosis. Then you can also have what? Then you can have candida, right? So HIV positive patient, they are generally asking questions. Remember, for meningitis they can even write you symptoms, you can identify the diagnosis, cryptococcal meningitis, right? इसका drug of choice पूछा था cryptococcal meningitis का. What is going to be the drug of choice? This is going to be liposomal amphotericin B. liposomal amphotericin B, right? Okay. In dysentery, what is dysentery? Dysentery will be what? Dysentery, you can also use what along with the flu cytosine. That is what you need to remember. We'll discuss this thing further, right? So next thing we are having is dysentery. Dysentery means bloody diarrhea, right? Bloody diarrhea. So when you have this bloody diarrhea, this is known as what? Dysentery. The most important cause for dysentery that we are having is going to be what? First one is Campylobacter jejuni, that can be due to uncooked or raw or uncooked meat can lead to this particular thing, right? It can be due to group B streptococci, that is going to be sorry. It can lead to what? GBS, which is going to be what? Guillain-Barré syndrome, and treatment we have already discussed, right? Okay, next one is entamoeba histolytica. Entamoeba is a it is going to be what? Amoeba, basically. Histolytica, histo means tissue, lytica means it is going to break the tissue, and these tissues are going to get broken, basically, right? So what is going to happen? You are going to have what? You are going to have bleeding, and this bleeding is due to what? Amoeba. So this amoeba can lead to what? Dysentery in the patient. This amoeba can enter into your liver, and it can cause what? Liver abscess. It can cause what? Liver abscess, right? And this amoeba is going to cause what? Liver abscess, right? Next one is your EHEC. This is going to be your enterohemorrhagic E. coli, and last one we are having is EIEC, which is going to be enteroinvasive E. coli, right? So these two can lead to what? Again, dysentery we have already seen, right? And Shigella is one of the most important cause for dysentery. This is going to have what? Shiga-like toxin, which is going to be causing what? Bacillary dysentery. What about the diarrhea we are having? The diarrhea we are having is due to Clostridium difficile. Clostridium difficile is going to cause what? Watery diarrhea. Pseudomembranous colitis you can have due to Clostridium difficile. Clostridium perfringens can cause what? Gas gangrene. Very important. You can use what? You can use hyperbaric oxygen. You can use 100% oxygen. Clostridium is an anaerobic bacteria. You can use oxygen. Along with that, you can also go for what? Amputation. This is what is going to be the treatment for gas gangrene or myonecrosis or flesh eating bacteria, we can also call it. Enterotoxigenic E. coli can also lead to diarrhea. This is your traveler's diarrhea. Protozoa that can cause diarrhea are going to be what? Giardia and Cryptosporidium parvum. Just now I told you this Cryptosporidium parvum in HIV positive patient, the size is going to be what? 4 to 8 micron, 4 to 6 or 4 to 8 micron is going to be the size. Vibrio cholera is going to be there, which is going to cause your rice watery stools, and viruses are going to be rotavirus and norovirus. I told you norovirus in the adults, rotavirus in the children's, right? And salmonella is going to cause what? Salmonella is going to cause what kind of diarrhea? This is going to be your pea soup diarrhea. So we are done with the bacteria in general, important parts of the bacteria in general we are done with, right? Are you guys understanding? सही है, समझ में आ रहा है? The next important thing is about the fungus. Next important thing is about the fungus. The first fungal infection we are having is going to be your pityriasis versicolor. Pityriasis का मतलब क्या होता है? Pityriasis rosea, pityriasis versicolor, pityriasis, multiple pityriasis we are having. Pityriasis means what? Scaling. Scaling means what? Accumulation of what? Epidermis or superficial layer, that is going to be stratum corneum, right? So scaling is going to be there. Versicolor means different color. Versi means different color. So you can have hyperpigmented or hypopigmented lesions are going to be there, right? So this is known as pityriasis versicolor. Scaling will be present, hyper and hypopigmented lesions on the back. This is known as pityriasis versicolor. This is known as pityriasis versicolor. यहाँ पर एक और डिजीज आप पढ़ते हो, that is known as pityriasis rosea. Remember rosea में red color lesion will be there, right? यहाँ पर कैसा है? Black and यहाँ पर hyperpigmented lesion है. Pityriasis rosea will be having red color lesion, और उस पैच को बोलते हैं mother patch, herald patch, and then you will be having Christmas fir tree pattern will be there, which is going to be the thing we are having, right? तो वो अलग disease है. This is going to be pityriasis versicolor, important है, right? यहाँ पर क्या क्या क्वेश्चन पूछे गए हैं? let me just jot down all the questions that has been asked previously. So Malassezia furbus, Malassezia globosa we are having, Malassezia furfur we are having. It is going to have hypo and hyperpigmented macules. Macules are going to be flat lesions, right? So you can have what? macules are going to be there, hyperpigmented, right? Pathogenesis में क्या होगा? This is going to be due to azelaic acid. This fungus is going to produce what? Azelaic acid, and this azelaic acid we are using in the creams also. Some people are using for the skin whitening, है ना? वो skin whitening cream आता है, उसमें क्या होता है? Azelaic acid will be there. This azelaic acid will destroy your melanin production and leading to what? Leading to whitening of the skin. तो ये जो fungus है, this fungus will also have the same property. It will produce this azelaic acid, and this azelaic acid will cause this particular whitening, and that is what is going to lead to what? Hypo and hyperpigmented patches on the back, right? So this is going to be what? Pityriasis versicolor, right? On KOH mount, sir, ये KOH mount क्यों करते हैं? We are going to take up this particular sample, putting on the slide, we are going to see it under microscope, right? So अगर hyphae या आपको you are going to find what? Yeast or hyphae, that means it is a fungal infection. अगर yeast या hyphae या कुछ भी नहीं दिख रहा, ये सब नहीं दिख रहा, KOH mount के अंदर अगर ये सब नहीं दिख रहा, that means it is not a fungal infection. To identify that fungal infection, we can make KOH mount. KOH mount कुछ नहीं है, we are going to take up the sample, scraping लेना है और slide पे डालना है. You are going to put potassium hydroxide, you are going to see it under microscope. This is known as KOH mount, right? So for pityriasis under KOH mount, what is going to be the thing? What is going to be the thing we are having? I'll take up your questions in the last, right? So what is going to be the thing we are having? KOH mount. KOH mount, you are going to get spaghetti and meatball appearance. What is the appearance you will be having? Spaghetti and meatball appearance. Very important question, multiple times asked question, right? So what appearance you are going to get on the KOH mount? Spaghetti and meatball appearance. Second important feature of this KOH mounting, always remember, it is going to do what? Keratolysis, है ना? जो केरेटिन के बोन्स है ना, उनको तोड़ देगा, right? So you can directly put this particular thing on the agar now. And this is the another question in the last exam. They have given you a case of what? Candida, and then they have asked you what is the agar we are going to use to grow candida, है ना? तो सारे फंगस में याद रखना कौन सा अगार यूज में लेंगे फंगस को ग्रो करने के लिए. दिस इज नोन एज सेप्रोड डेक्सट्रोज अगार और SDA is the agar we are going to use. So under SDA, when you are going to put the same thing, KOH mount sample को आप जब SDA में डालोगे, you need to put some olive oil here. You are going to get appearance. This appearance is known as what? Fried egg colonies. This appearance is known as fried egg colonies. These are the fried egg colonies that you are going to appear. You are going to get. This is going to be your diagnosis. This is how you can identify fried egg appearance. और कहाँ पढ़ते हो बेटा? Where else you study fried egg appearance? One is in the mycoplasma. One is in the mycoplasma. In Papilo agar, you are going to get this fried egg appearance. Another one in the pathology, oligodendroglioma, non-which tumor. Oligodendroglioma will also have this fried egg appearance, right? तो ये तीन जगह fried egg appearance पढ़ते हो, right? The first one is going to be under SDA in pityriasis versicolor. You are going to get this fried egg appearance. ये spaghetti and meatball appearance is going to be there. Treatment क्या करना है? वैसे तो asymptomatic है, कोई दिक्कत वाली बात नहीं है, but still if you want, you can use what? Antifungals like clotrimazole or ketoconazole. You can apply. This is how you are going to get rid of this particular thing, right? So ultimate understanding, this thing is a commonly happening in young individuals. On the trunk, you will be having hyperpigmented lesions, and this hyperpigmented lesions are due to what? Azelaic acid, right? You can put on the SDA, you can find spaghetti meatball appearance. On the on the KOH mount, you will find a spaghetti and meatball appearance. On the SDA, you will find what? All this is going to be your fried egg colonies. Where else you will get the fried egg colonies? This is also important. You can remember. You can get fried egg colonies in the mycoplasma and oligodendroglioma, right? Next thing is going to be what? Tinea infection. Tinea infection of the head is going to be known as tinea capitis. सिर में अगर fungus infection हो गया तो tinea capitis बोलेंगे. Scalp और hair में, right? And this fungus is going to feed on the oil. Oil लगाना है नहीं लगाना है? Remember बेटा, fungus is going to feed on the oil that you are applying. जो अपन तेल लगाते हैं उसमें fungus feed करता है. Fungus do not want, fungus loves the moisture, right? हमको moisture नहीं रखना. We need not to have the moisture, right? This oil will convert into your oleic acid, and that is going to be the food for the fungus, right? So someone is having fungus in the head, right? Fungus means this is the same fungus which is going to cause what? Dandruff in the head, right? So malassezia can cause dandruff, right? So you need not to apply the oil, right? Basic understanding, right? What is going to be the thing we are having? We are having tinea capitis. Tinea capitis, two important entities you need to remember. One is your favors, another one is your carrion. One is your favors, another one is your this is going to be your carrion, right? Favors is going to be due to tinea stoloni. right? It is due to tinea snolini, where you will be having crusting, dense crusting is going to be there. This is going to cause what? Dense crusting is going to be there. This is going to be what? Fevers, right? You can have dense crusting. Whereas this chorion is going to be inflammatory type of boggy swelling is going to be there. This is going to cause what? Boggy swelling. So whenever a kid is coming to you with this type of boggy swelling, this is going to be due to what? This is due to tinea mentagrophyte, or one more organism you can remember, that is going to be tinea verrucosum. This is going to be tinea verrucosum or tinea mentagrophyte are the organisms that can lead to what? This chorion formation, right? This is a chorion and this is the fevers, right? Both the tinea you are going to use the drug of choice. This is going to be what? Griseofulvin, right? Tinea capitis में drug of choice griseofulvin है. बाकी सब tinea में you can give terbinafine, right? You can have tinea infection on the beard. This is known as tinea barbae. You can have it on the body. Corporis मतलब body. Cruris means hidden areas. These are going to be your groin or inner thighs, right? So these are the tinea cruris, pedis, and corporis, depending on the location we call it, right? And the drug of choice for these particular one is going to be what? The drug of choice for these one is going to be what? Terbinafine. Terbinafine, right? The next thing we are having is subcutaneous mycosis, where you will be having the first one. This is going to be first one is going to be what? M for mycetoma. So this is the mycetoma we are having. How many types of mycetoma you can appreciate? How many types of mycetoma do we have? Yes, treatment we can give amorolapine also. We can give multiple things we are having. Cyclopirox we are having, efavirenz we are having, right? Tevaborol we are having, multiple things we can use, multiple drugs we are having, but just the drug of choice we are writing, right? So this is your mycetoma or Madrella foot we are having. We can have U mycetoma, we can have actinomycetoma, right? This U mycetoma is going to be a fungal infection, whereas actinomycetoma is going to be a bacterial infection. Whenever you are finding something is coming out from a sinus, something is coming out, you need to think about what? Mycetoma, right? In the legs of a farmer, something is coming out from the legs. This is going to be mycetoma. Remember, mycetoma, if it is a fungal infection, after E there will be F. And actinomycetoma, if it is a bacterial infection, you can give antibiotics. You can use the Welsh regimen. You can use cotrimoxazole, which is a combination of trimethoprim plus sulfamethoxazole. You can also use what? Ampicillin, right? So this is where we are going to use the Welsh regimen. Whereas in this particular case, you can use antifungals, but generally fungal infections are going to be what? Sidal. Fungal infections are going to be what? Chronic, right? And if they are going to be chronic, generally they are going to be causing more damage, leading to what? We need to go for what? Amputation generally. We need to go for what? Amputation generally. So fungal, we need to go for what? Amputation, right? This is going to be what? Which appearance you are going to appreciate? This is also known as chromoblastomycosis, beta. Chromo means color, blastomycosis is a fungal infection. So this is going to be a colorful, warty, dematitious fungal infection that is going to happen on the foot also, right? So this thing is going to be there. What are these bodies? When you are going to take out this sample, you are going to take the scrapping, you are going to put it on the slide, you are going to find these bodies. These bodies names are important. What are the names of these bodies? These bodies are known as copper penny body. Everybody remembers this, but other names are also important. They have asked all four names, right? So copper penny bodies, these are also known as medlar bodies. Speak wherever you are. They are also known as medlar bodies. They are also known as murilliform bodies. They are also known as sclerotic bodies. Sclerotic bodies. So these all names has been asked previously, right? Copper penny, medlars, murilliform, and sclerotic. The drug of choice we are going to give is itraconazole, but the treatment remains a long time treatment, right? We can debride the area also. We can give this itraconazole in this particular case, right? Next one, we are having this lymphocutaneous disease, which is going to be involving the lymph node root as well as the cutaneous infection. That is why it is a lymphocutaneous disease. This is also known as Valentine disease. This is also known as Valentine disease. By now, I hope you get the name right? This is going to be your sporotrichosis. Sporotrichosis is going to be the disease that we are having, right? Here, what are the things we are having in this porotrichosis? You will be having what? You will be having you will be having due to thorn pick injury, Rose Gardner's disease. This is also known as Rose Gardner's. You are going to have what? Thorn pick. This is also known as a Valentine disease. After that, you will start to have development of what? Lymphocutaneous lesion, right? And this lesion will be moving in this particular fashion. ये ऐसा ही दिखेगा. Lymph node का रास्ता है, उसके ऊपर cutaneous skin manifestation. You will be having what? Nodulo ulcerative lesions are going to be there. Nodules means they are going to go deeper. Ulcer means they are going to involve epidermis as well as dermis, right? So this type of lesions are going to be there, and these lesions are going to be there, right? So this is known as Rose Gardner's disease. और क्या क्या पता है Rose Gardner's के बारे में? What kind of sporulation is going to be there? It is a dimorphic fungus, so you will be having flower like sporulation will be there. Sporulation, remember the story, है ना? कैसा yeast होता है इसका? cigar shape yeast, right? budding कैसा होगा? narrow based budding. अभी बताता हूँ कैसे याद करना है. narrow based budding is going to be there, right? and बाइक कौन सी है? splendor hopefully. which phenomena you are going to see? splendor hopefully phenomena. And which bodies are going to be there? Asteroid bodies. Treatment is going to be again itraconazole you can use, right? So there is a boy who's going to propose someone, right? And on the Valentine day, and with the rose, right? And he's taking these flowers, and the uncle is a narrow-minded uncle. He is smoking the cigar, right? He went to the father of this particular girl, right? The uncle is a narrow-minded uncle. He's smoking the cigar, and he's going on a bike like Splendor, and the uncle kick him out to the asteroid, right? Something like that we have met, right? So mostly points are covered here, right? This is going to be what? This is going to be a Rose Gardner's disease. This is going to be the condition of the patient, है ना? ऐसा हो गया था, इतना मारा अंकल ने उसको, right? This pink eosinophilic material, right? You can appreciate this is the yeast which is surrounded by this pink eosinophilic material. Eosinophil मतलब होता है pink color का material, right? So this pink eosinophilic material is going to be there, surrounded by this yeast is surrounded by this material. This body is known as what? Asteroid body, right? Asteroid body बोलते हैं इसको, right? Another thing we are having is going to be what? This particular entity. What is this entity? नाक को क्या बोलते हैं बेटा? नाक. Nose is also known as rhino, right? This is a disease. This is known as rhinosporidiosis, right? Is it rhinosporidiosis? Is it rhinophyma? Is it rhinoscleroma? What is this particular disease? क्या बोलेंगे इसको? So this is rhinosporidiosis, which is due to Rhinosporidium seabury. Right, ये बंदा नहाने गया था, and now this mass is coming throughout his nose, right? This is going to be what? Due to Rhinosporidium seabury, right? What is the treatment would you like to do in this particular case, and what is the drug would you can use, right? So drug हम कौन सी यूज में ले सकते हैं? हमने क्या पढ़ा था? डेप्सोन हम लोग इस पेशेंट को दे सकते हैं, and the treatment is going to be what? Functional endoscopic sinus surgery we can go for, right? We can go for this first surgery, right? Remember वो दूसरा है rhinoscleroma. Rhino means nose. स्क्लेरोमा मतलब होता है हार्डनिंग. स्क्लेरोमा मतलब होता है हार्डनिंग. स्क्लेरोसिस होता है ना, आर्टरी एथेरोस्क्लेरोसिस. तो हार्डनिंग इज गोइंग टू बी देयर. दैट इज गोइंग टू बी अनदर डिजीज दैट इज नोन एज वुडी नोज. वुडी नोज, दैट इज नोन एज वुडी नोज. वुडी नोज पे भी क्वेश्चन आता है. दे विल आस्क यू फ्रॉम ईएनटी व्हाट काइंड ऑफ बॉडीज यू आर गोइंग टू फाइंड इन द वुडी नोज. सो रिमेंबर यू आर गोइंग टू फाइंड व्हाट? मिकुलिथ सेल्स एंड रसल बॉडीज. मिकुलिथ सेल्स एंड रसल बॉडीज आर फीचर ऑफ व्हाट? राइनोस्क्लेरोमा, है ना? जब आप हिस्टोपैथोलॉजी लोगे, आप सैंपल लोगे फ्रॉम द नोज, यू आर गोइंग टू टेक आउट द एचपी एग्जामिनेशन सैंपल, यू आर गोइंग टू फाइंड व्हाट? मिकुलिथ सेल्स एंड रसल बॉडीज आर गोइंग टू बी देयर, राइट? So this is going to be rhinoscleroma, rhinosporidiosis. And one more entity that you need to remember is rhinophyma. Phyma मतलब होता है potato like tumor, right? So rhino means nose, and you will be having what? Hypertrophy of ectopic sebaceous gland. Hypertrophy will be there. Sebaceous gland hypertrophy. This is known as what? This is known as rhinophyma, right? Rhinophyma also known as potato nose. Potato nose, right? So that is a different entity. You just need to remember these things, right? सर इसका HPE करेंगे तो क्या मिलेगा? Here you will be finding endospores with spherules. with spherules. एक आधा क्वेश्चन इनमें से पूछ लेते हैं because you are tend to get confused in this particular area, so we are going to make it strong, है ना. अब तो एग्जाम आ गया हमारे, स्ट्रॉन्ग होना चाहिए. राइनोस्पोरिडियोसिस पूछा, राइनोस्पोरिडियम सीवेरी, डेप्सोन दे सकते हैं सर, फेस कर देंगे. राइनोस्क्लेरोमा पूछा, स्क्लेरोमा में वुडी नोज, स्किन हार्डनिंग विल बी देयर. Rhinoscleromatis is the bacteria, Klebsiella, right? That is the bacteria that is involved here, right? And this is going to have what? Miplis cells and Russell bodies. What about this rhinophyma? It is going to be sebaceous gland hypertrophy, potato nose can be there, right? So these are the subcutaneous fungus that we are having, right? Now we are having this dimorphic fungus, and this dimorphic fungus we are having the first one. The yeast cell is going to be there. Can you appreciate? This is the yeast cell inside the cells, right? This is going to be intracellular yeast identification. This is only feature of histoplasmosis. Histoplasmosis. This is also known as High disease. High say you can remember Ohio disease. This is also known as Darling disease. This is also known as Caves disease. Let's go to cave. Hi darling, let's go to cave, like that you can remember Caves disease, right? What are the things this person is going to have? This person is going to have all the inhalation of these spores that would lead to what? Pancytopenia. That would lead to what? Pancytopenia is going to be there. Just a minute. That would lead to pancytopenia, right? So everything is going to get decreased in the body. WBCs, RBCs, platelets, everything are going to get decreased. This is pancytopenia. You can appreciate these cells are going to be which cells? ये कौन से हैं? ये पेलर 1/3 वाले cells कौन से हैं? These are going to be your RBCs, right? These are going to be your RBCs. Here you will be finding histoplasmosis. You are going to find what? Macrophages. These bigger cells are going to be what? These are going to be your macrophages. And what you can appreciate here, the inside the macrophages we are finding these round yeast cells. This is a true yeast, right? So you are going to find this yeast inside macrophage. Yeast inside macrophage. What kind of symptom this person will be having? TB like symptoms are going to be there. TB like symptoms. So this is a case of what? This is a case of histoplasmosis, right? Here you will be finding narrow based budding, है ना? वो अंकल भी narrow था, sporotrichosis वाला also has narrow based budding. This one is also having narrow based budding, right? This is going to be what? This particular one is going to be what? This particular one is going to be your blastomycosis. This is your blastomycosis. This blastomycosis will be having what? You can appreciate this bee is looking like 8. ये बी कैसा दिख रहा है बेटा? 8 जैसा दिख रहा है. This bee is looking like 8. This particular one is going to have figure of what appearance? Figure of, अगर इसका इमेज पूछे, figure of 8 appearance will be there, right? This disease is also known as North American disease. इसका दूसरा नाम क्या है? North American और Gilchrist disease. North American और Gilchrist और Chicago disease. Just remember important thing about this particular one. B for blastomycosis, it is going to have what? Broad based budding. Very good, very good, very good, Ruby, right? Broad based budding is going to be there. Perfect, right? So this is going to have what? Broad based budding is going to be there, right? As we have already discussed, right? This is the mnemonic that we have studied is going to be what? For dimorphic fungus, HSBC P square, right? So H for histoplasmosis we are done. As for sporotrichosis we are already done sporotrichosis, right? B for blastomycosis. C for this is going to be what? This is going to be your coccidiomycosis, right? And this is going to cause what? Desert rheumatism, right? This is going to cause what? Desert rheumatism, also known as San Joaquin Valley fever, also known as San Joaquin Valley fever, right? You can appreciate this particular thing. I told you endospores with this ferrule. So this particular one is going to be what? This particular one is ferrule, and here these small yeast cells are going to become what? Endospores. So this is going to be the appearance that we are having. but the important appearance they are asking is about these three one, right? Next one is going to be this particular one. What you can appreciate here, what it is looking like, कैसा दिख रहा है ये? This one is known as what? This one is known as paracoccidiomycosis. Also known as South American disease. This is also known as South American disease. This is known as paracoccidiomycosis, also known as South American disease, and the appearance that you are going to get is known as what? Mickey Mouse appearance. The appearance is Mickey Mouse appearance, also known as Mariner wheel appearance, also known as pilot wheel appearance. Right? So these are the appearances that we are going to get in this particular one. This is going to be the paracoccidiomycosis, and the last one is going to be P square, that is going to be your penicillinosis. Penicillinosis, also known as telleromycosis. Right? Here you will be finding this brick red color pigment. You can appreciate brick red color pigment. Just remember the image. Along with that, you are going to have these microconidias. These conidias are going to be there, right? Which are going to give you this broomstick like appearance, right? This is going to be your broomstick like appearance, jhadu jaisi appearance. Conidial microconidias, these are going to give you broomstick like appearance. And this disease is generally happening in HIV positive individuals, very commonly seen in the HIV positive individuals, right? So these are your HSBC P square, HSBC P square, right? The next thing we are having is about what? This is going to be your infection that we are having. This is going to be your Candida oral thrush that we are going to appreciate. This is your oral thrush, Candida infection or oral thrush that we are having. And this appearance is very important. This appearance is known as what? Here what is happening, from the mother cell something is coming out. From the mother cell something is coming out. This is known as germ tube formation. This is also known as a phenomena. This phenomena is known as Raynaud-Broad phenomena. This phenomena is known as Raynaud-Broad phenomena. This phenomenon is known as Raynaud-Broad phenomena or germ tube test formation, right? Here also you are going to find chlamydospores on the corn meal agar, right? So this is the important thing. What about this particular one that we are having? This is going to be your cryptococcal capsule. These are going to be your cryptococcal capsule, and this Cryptococcus neoformis is the most important cause for HIV positive patient are going to have what? Meningitis. So meningitis in HIV positive individual, the answer will be what? HIV positive individuals suffering with the meningitis, the answer will be what? Cryptococcal meningitis, right? Another question they have asked you, how to identify this particular one with the help of stains, and these stains are going to be your nigrosin or India ink. Examiner has asked the question about India ink staining, right? So what kind of stain we are going to use? India ink or nigrosin stain we can use to identify this particular one, right? On biopsy, this is going to be CSF sample. This is going to be the sample of CSF. Meningitis patient, we are going to take the CSF sample, है ना? Question में कहीं भी CSF लिखा हुआ हो, think about meningitis. कब निकालेंगे CSF? Meningitis को identify करने के लिए. On biopsy, you are going to find these are the capsules. ये जो दिख रहे हैं आपको, these are the capsule. It is a capsulated fungus, right? So these are the capsule, and the stain that we are using is mucicarmine. The stain that we are using is mucicarmine. Here you can identify some shiny appearance is going to be there, right? And this shiny appearance is going to be due to calcoflower white. Remember, calcoflower white is the stain for fungus, right? And you can identify these capsules are going to be visible, right? What are these capsules? These capsules are the capsules of cryptococcus, right? These are the cryptococcal capsules that you can appreciate, and this is the fluorescent dye that we are going to use. Fluorophore, you can call this chemical as a fluorophore. This cryptococcus is very important. What is the treatment? The treatment is liposomal amphotericin B. Liposomal amphotericin B, right? Along with that, you need to mark your answer if flu cytosine is there in the option, you definitely need to mark this particular one, right? ये वाला आंसर सेलेक्ट करना है. If the patient, if in the question it is written liposomal amphotericin B plus flu cytosine, please do mark this one, because the other options are also going to be there, which are going to have only liposomal amphotericin B, or maybe amphotericin B they can pair with the flu cytosine. So remember liposomal amphotericin B along with the flu cytosine, right? You can also do a latex agglutination test, latex card agglutination test to identify what? These cryptococcal capsules, right? So these are the important points about the cryptococcus that you need to remember, right? The next one we are having is about this particular one. Here you can identify this structure is getting black, this area is getting black, this is a dead necrotic mass that we are having, this is going to be the dead necrotic mass that we are having. This is a case of what? This is a case of mucormycosis. This is a case of mucormycosis, right? How you will identify? This is also known as rhinocerebral mycosis. How this is traveling? It is traveling through the nose. It will come to the eyes via cribriform plate, and then through the it will come into your nose, and via the lamina pepracia it will enter into your eyes. It will come into your nose via lamina pepracia, which is the thinnest layer, this medial side. It will enter into your eyes, and then it will go to the brain with the help of cribriform plate, right? So that is why it is known as rhino, from the nose to the cerebrum, right? This is known as rhinocerebral zygomycosis, right? So this is going to be your mucormycosis. For mucormycosis, what you need to remember, you are going to take out the sample. When you are going to put the sample on the KOH mount, you are going to take out the sample for diagnosis. You will take out the sample, you will destroy the dead necrotic mass, you will do the debridement. Very important thing is debridement is very important. Even before giving the drug of choice, you need to do surgical debridement. तो ऑप्शन में अगर लिखा हुआ है debridement, so first thing you are going to do is going to be what? Surgical debridement. Because अगर यहाँ पर blood supply नहीं है, you will give drug IV, it will not reach to the effector organ, right? So that is why we need to remove this black necrotic mass, and then you need to go for what? Then you need to go for what? Liposomal amphotericin B. So first thing is going to be debridement. Post debridement, you are going to take out some sample and you are going to put it on the QH mount, right? When you will put it on the QH mount, you are going to find something like that, and this particular one is going to have what? This particular one is going to have what? Wide angle. You can appreciate this angle is going to be acute or obtuse. Obtuse means about 90. So this angle is going to be obtuse angle is going to be there. You will be finding these hyphae are going to be there, right? These hyphae are going to be septate or aseptate. These hyphae are going to be aseptate hyphae with obtuse angle. This is how we will understand this is a case of what? Mucormycosis, generally happening in immunocompromised individuals which are going to have what? Decreased in immunity, or diabetes mellitus patient, right? These are the people who are going to have more sugar, people who are having what? More risk of infection. This is going to be what? Mucormycosis, right? The treatment of choice for this mucormycosis is going to be, first thing is surgical debridement, then you go for what? Liposomal amphotericin B, right? You can also use some oral drugs, and the only two important oral drugs you can remember is going to be what? Is savuconazole and posaconazole. Isavu and posaconazole are the two oral drugs that you can also use along with this particular mucormycosis, right? The next entity we are having is going to be what? The one with the acute branching. Can you appreciate? This is with the acute branching, right? Acute branching is happening, and that type of branching where one branch is giving rise to two other branches, this branching is known as dichotomous branching, right? So acute angle is going to be there. Acute angle is going to be there. Septate hyphae is going to be there. Acute angle with septate hyphae along with the dichotomous branching. This branching is known as dichotomous branching. So these things are going to be coming together. This is going to go in the favor of what? This is a case of aspergillus, right? So aspergillus, you can remember acute angle septate hyphae. From the name itself you can also remember. So whenever you will find the acute angle and septate hyphae, this is going to go in the favor of what? Aspergillus. And if it is going to have wide angle, this is obtuse angle, this is going to go in the favor of what? Obtuse angle is going to go in the favor of mucormycosis, mucormycosis, right? And this appearance that we are going to get, this appearance is known as what? ping pong ball appearance is going to be there. This appearance is known as what? Ping pong ball appearance, right? This ping pong ball appearance you are going to find, and this is going to be a case of what? This ping pong ball appearance is a case you will be finding in pneumocystis cerevisiae pneumonia. You will be finding in pneumocystis cerevisiae pneumonia. This is something you will be finding in pneumocystis cerevisiae pneumonia, right? What other things you will be finding here? Multiple things you can find. It is an important entity, right? Whenever you are going to take out, this person will be coming to you with the symptoms of pneumonia. When you will take out the sample, which sample? Bronchoalveolar lavage you are going to take out. In BAL, bronchoalveolar lavage, you are going to find, you are going to put the stain. The stain is going to be your gomori methionine silver stain, right? When you will put this silver stain, you are going to get this black color appearance. We already know silver stain is going to give you black color appearance, and you will be finding these are the yeast cell in what sample? In the bronchoalveolar lavage, which are causing what? Which are causing the pneumonia in the patient, which is a HIV positive patient, and the CD4 count is low. CD4 count is 200, right? So this is the patient which is a HIV positive patient having CD4 count around 200, having pneumonia, and we are going for bronchoalveolar lavage. We are entering through the bronchus, we are doing endoscopy, right? And we are going to take out this sample. We are going to put this sample under GMS. You are going to get what? Ping pong ball appearance is going to be there, and this is going to give you a confirmed diagnosis of what? This is going to be a case of what? This is the case of pneumocystis cerevisiae pneumonia. You can also go for CT or X-rays. You are going to find what? Miliary mottling. Miliary mottling is going to be there, right? So here you will be finding what? Miliary mottling along with the perihilar opacities, right? Whenever you are going to have any infection in any area of the body, what will happen? The nearby lymph node will get swollen, right? So what will happen? In the TB also you will be having perihilar lymphadenopathy. Here also you will be having what? Perihilar lymphadenopathy. Around the hilum you will be having lymph node, they will get bigger because of the infection, right? So you can have miliary mottling with perihilar lymphadenopathy, right? Along with that you can have this particular one. How to treat, sir? How to treat? For treatment you are going to give what? Again the same drug you are going to give. This is going to be what? Combination of trimethoprim plus sulfamethoxazole, cotrimoxazole. So this is going to be the drug of choice for this particular type of pneumonia, right? So this is going to be the pneumonia. This is going to be the thing we are having, right? I think we are running short of time today, right? And please try to do the revision, right? We have to end up this session right now, right? So you will be having another class in next few minutes, right? If you find any difficulties, you can always connect back to me. I will share the annotated PDF with you. All the very best for your examinations, right? If you find any difficulties, any doubts, you can feel free to reach out to us. No issues with that, right? All the very best for your examination. Thank you so much for all the patience. If you are having any doubts, you can ask me right now. Few part is left that you can rewatch through the notes, or I will share this particular one, right? It was great teaching you guys. Lovely teaching you. Take care, guys.